So I think you are live, and we can start. I'm Thiago Cunha. I'm a professor of pharmacology in University of São Paulo in Ribeirão Preto. It's my great pleasure to be here this afternoon and to present our next speaker, Professor Carmen Feijoo. First of all, I'd like to thank Carmen that accepted the invitation of the organizers to be here today and give a talk. So Carmen, she's a full professor in University Andrés Bello in Chile. She did graduate PhD in Chile and also in Spain in Autónoma University of Madrid. And she also did a postdoc in Chile University, and then she started her career as an independent researcher, University of Andrés Bello. Her interested in research is mainly on mucosal immune response, especially to understand myeloid cells and regulation of this in terms of communication with microbiota, which is very interesting at this moment. So, and she has mentored several PhD students and has been published very important papers in important journals. So thank you very much, Carmen, and please take your time to give this excellent talk. Thank you very much. If George, if you want to say some words, please be free.
Well, just a very brief thing. So I'm Jorge Galindo Villegas, and it's a pleasure for me to be here. I'm really excited to be hosting this, co-hosting this session. Carmen is one of the best examples that we have that in Latin America, it's also possible to do good things because as Thiago already mentioned, she has the opportunity to be based in Europe or in America and some other places, but she decided to study in Chile, and her contributions to the science using this model, the Zebrafish, has been really, really remarkable. So then let's put attention on what she has to say today. So Carmen, please, the floor is yours.
Thank you, Jorge. Thank you, Thiago. Yeah. Now I want to share my screen. Please tell me if you see my presentation. Yes?
Not yet. Now it's coming. Yeah, now it's here.
Now?
Yeah.
Yeah.
Yeah, perfect.
Thank you. Oh, well, thanks to the organizer for inviting me, Jorge and Yves. And today I'm going to talk about the intestinal immune response that decide if the intestine induce a tolerogenic response or induce an inflammation. We know that the intestine is exposed to a lot of different antigens, and in the case of pathogens, when different types of pathogens ingress to the intestine, this induce an intestinal inflammation. But how this works? Okay, we know that mononuclear phagocytes, mainly macrophages or dendritic cells, they detect harmful antigens, secrete signals, and induce a pro-inflammatory response to establish the inflammation and protect the body. But let's think about food. What happens with food? Because there are all of these foreign antigens, and let's concentrate in protein or stick in a barbecue. These are foreign antigens, and they ingress to the gut, and they don't induce inflammation, but rather homeostasis is maintained. So how is this happens? Again, mononuclear phagocytes, macrophages, detect this stick, the proteins, secrete signals, induce the tolerogenic response, and maintain homeostasis. So the question is, how can macrophages do this? How can they distinguish between the signals and induce very different, indeed opposite response? This is very important because, allergy or autoimmune response, or we need to maintain a homeostasis in the gut. So to be sure that the correct antigen will be detected as a tolerogenic antigen and induce this side, the tolerogenic response is very important. So here now, in this talk, I want to share with you what we have been done regarding macrophages and these two processes. And for this, we use it two strategy. This is a feeding strategy that we established in the lab several years ago, and we have some control diet that feeding the fish with this diet, we induce oral tolerance, a tolerogenic response. We fed the fish for more or less than 10 days with the regular feed, and then we switched to this control diet. And after 8 days, we made the analysis. We took the samples to make the analysis. And if we look at the transcription level of different cytokines important for inducing oral tolerance or inflammation, we can see that the transcription level of FOXP3 and interleukin 10 are higher after the feeding strategy compared to the situation before feeding, suggesting that more tolerogenic condition is establishing. And regarding the pro-inflammatory cytokines like interleukin 17 and interleukin 12, they are not induced in this condition. If we see the amount of myeloid cells, we see no change. So the diet didn't change the number of neutrophils and macrophages, again pointing to a situation where no inflammation is induced. And lymphocyte, they increase. Also the transcription level related to them, and we think that is because of the increase in the number of regulatory T cells, but this is a thing we need to check. So we did also another test to verify that we have induced this oral tolerance, and this is we fed fish with a control diet, but also one of the group we include also ovalbumin, and a couple of days after the feeding strategy, we microinjected this ova in the muscles and see if this induce an inflammatory response by quantifying the amount of neutrophils that were recruited to the site of microinjection. And as you can see here, those fish that were just fed with the control diet and then microinjected it over induced the inflammation, but those that were fed with the diet and over did not induce the recruitment of neutrophils, so the inflammation, after microinjecting ova in the muscles, indicating that in this fish, the oral tolerance was induced not only in the intestine but also systemic, as is expected. And here, ova was recognized as a foreign antigen. So we are now in the condition where fish had oral tolerance in the intestine. So regarding macrophages, what we have seen here, this is a region of the gut. You can see it surrounded by or delimited by the black pigments, and in green you see the macrophages. This is a transgenic MPEG-1 GFP line, and as you can see here in this video, we can see two types of cells, some that are more round and another that are very big with long protrusions, more stellate cells. So in order to quantify and define the amount of each population in the gut, we choose these two parameters, circularity and the ferret diameter, to define which cell is a round macrophages and which is a stellate one. And after defining the parameters for each population, we quantified the amount of round macrophages and stellate macrophages in the gut. And as you can see here, we have much more stellate macrophages, more than the double, than rounded macrophages. But yeah, we are talking about antigen recognition, so we need to detect and take up these antigens. So for analyzing this aspect, we microinjecting fish that were fed or that were fed with the control diet, so they are in the tolerogenic condition with ova. We did this microgavage with ova that is red fluorescent, you will see it, and we analyzed it if the rounded macrophages and or the stellate macrophages catch it, take up ova, and just to show you what happens. If you see here again, you can see how a macrophage take up this ova and catch it. So we quantified that, and you remember that stellate macrophages were at least 2 or 3 times more than rounded macrophages, but in antigen uptake is the opposite around. Rounded macrophages are mainly the cell type here that are taking up antigen. Near to 40% of macrophages, rounded macrophages, are taking up antigen. So, okay, we know now that rounded macrophages take more antigen than stellate macrophages, but what happens with antigen processing? After catching antigen, we need to process antigen to present it to T cells. So to analyze this, we use it DQ ova, that is an oval albumin that is labeled fluorescently, but when ova is intact, fluorescence is quenched. So only after the protein is processed, so we have these small peptides, we see fluorescence. So we see something like this. here we just see some cells with ova, DQ ova fluorescent, and this indicate that these cells have not only catch the antigen but also processes it. And what we have seen is that again both cells process the antigen, but of course more rounded macrophages process the antigen because more rounded macrophages take up antigen. We also blocked the antigen process with chloroquine. This is a chemical compound that block the enzymes that process the protein. So what we expect is no cell with fluorescence in the cytoplasm. So, and this is what we obtained, indicated that our results were correct. So what do we have in the intestine in the heterogenic condition? These two populations, the stellate macrophages take up antigens in less percentage than rounded macrophages. Both process it, but rounded macrophages do it in a higher rate. And with this, they induce the tolerogenic or send signals to induce the tolerogenic response and maintain homeostasis. But what happens now in the inflammatory condition? Here we introduce another diet, an inflammatory diet. So we fed fish with regular diet for the 10 days, and then we choose to feed with the inflammatory diet or the control diet. We have these two groups, and then we process the samples for analyzing the transcriptional levels. And as you can see here, we have these two conditions. The light green is tolerance, and the dark gray, gray is inflammation. And pro-inflammatory cytokines, interleukin 17 and interleukin 22, are higher in the inflammatory condition, and FOXP3 and interleukin 10 are higher in the tolerogenic condition. So suggesting that we induced both conditions. In the number of neutrophils are higher in the inflammatory condition, confirming that we have this inflammation, and also the number of macrophages. Regarding the two populations, you see we have both, again, the stellate macrophages and the rounded one. And as in the tolerogenic condition, the stellate macrophages are much more than the rounded macrophages. But interesting, stellate macrophages in the inflammatory condition increased even more than in the tolerogenic situation. And as I don't know if you saw in the timelapse, I will play it again. Here you see that some stellate macrophages are recruited from outside. So these are something that we are analyzing, but we think that this increase in stellate macrophages is due to a recruitment in stellate macrophages from outside the gut. Another interesting question that we posed is that are macrophages activated cells? And with activated, we mean, are they pro-inflammatory? Are they expressing TNF-alpha, very important marker for pro-inflammatory macrophages. And as you can see here in the graphic, in the quantification, in the tolerogenic condition, we don't have macrophages nor rounded nor stellate, neither stellate, that express TNF alpha. But in the inflammatory condition, some of the stellate macrophages express TNF alpha. And if you see this time lapse again, here, macrophages, the red one, are the macrophages, and TNF alpha is in green, here, in green. And this area, the one to the right, is the area of the gut where antigens are taken up, are catched. So, and we see that the TNF alpha positive macrophages are more to the left of the video, and we think that they are not participating in the antigen catching, but doing something else. So this is also something that we are looking at the moment. So what happens with antigen uptake? Here in you can see again we did this the microgavage with OVA, and we quantified the number of rounded and stellate macrophages that have these red vesicles in the cytoplasms, and again, a higher percentage of rounded macrophages catch antigen more than the stellate macrophages. But what is interesting is that the amount or the percentage of rounded macrophages that take up antigen in the inflammatory condition is less than in the tolerogenic one. Suggesting that there is another cell type that is responsible for antigen uptake during the inflammatory condition. We analyzed it if maybe dendritic cells could be a cell type that participate, and the answer is yes, but not in a higher degree. As you can see here, very few dendritic cells take up antigen in the tolerogenic condition, and the number of cells taking up antigen increased during inflammation, but not much. So we have macrophages and dendritic cells taking up antigen in the inflammatory condition, but we suspect that there is another cell type that may be involved. Maybe just as in mammals, using the epithelium as a tool to introduce these antigens and get it to immune cells. Okay, but now back to antigen uptake by macrophages and processing. We analyzed what happens now in the inflammatory condition, and as you see, the situation is very similar to that of antigen uptake. So processing, both population process antigen, but in a few, just few cells in stellate macrophages, a little more in rounded macrophages, and rounded macrophages in less percentage that in the tolerogenic condition. So, in summary, what I share with you today is in the intestine, we can identify these two types of macrophages, rounded and stellate one, and both are present in the tolerogenic condition and during an inflammatory process. Um, in both conditions, stellate macrophages are in a higher amount that rounded macrophage, but during inflammation, these stellate macrophages increase even more. And we think that this is because of the um, recruitment from the outside. We also think that these pro-inflammatory macrophages are not taking part in antigen uptake, but doing other process or participating in other process in the gut. And importantly, rounded macrophages are in charge, or mainly in charge of antigen uptake during or in the tolerogenic condition. But in the inflammatory situation, we think that it should another cell type, the one that are in charge of taking up antigen and processing it. So what we are doing now is we already separate these two populations, and we are doing single cell RNA-seq to analyze the populations that we have and the gene expression here. So, and with this, I want to thank the person in the lab and Yalen del Rio. She's a PhD student in the lab. She did most of the work, and also Catherine Astorga some of the work with the dendritic cells. Thank you.
Thank you, Carmen. Thank you for the talk. Very interesting. And we are open for question. I will see if we have a question in the chat. There is one from Anna Lucchiari. She wanna know, do the macrophage migrate? These macrophage, do they migrate to other tissue after phagocytosis? So there's a traffic of these macrophages, and how about the effect on the brain?
Yeah, what we have seen is, yes, we see macrophages that are positive for OVA migrating outside the gut. But I don't know if they just are going outside the gut to reach other tissues, but because we have not quantified yet this, but I think that there are going in and out from the gut. I don't know why, but not that they migrate to other tissues. This is what we have seen for now.
I have a question. Have you tested for the loss of tolerance in the inflammatory model, or you didn't have any?
You mean changing from
have you challenged the animals before or after, as you did at the beginning? To see if there is a loss of tolerance.
Yeah,
in
we can do that, changing the most important thing that we have to establish is the cytokine situation, no, in the gut. So to establish the tolerogenic or the inflammatory condition, we need the typical cytokine for each condition. So if we have the tolerogenic condition and we change the diet from the control diet to the inflammatory diet, we will induce this inflammation, and we can see if again microinjecting OVA.
Yes.
Yeah, this is a good idea. No, we haven't do it, and but I expect that OVA will be recognized as a not harmful antigen, because um, when you have some intestinal inflammation, it's not that all the food that you eat induce inflammation. So this is something that is not that didn't change because you have inflammation. This is what I expect, but no, we haven't do the experiment.
Uh, from the audience, there is no more questions. Georgie, do you want to?
Yes, sure. If there are no more questions from the audience, then I would like to ask something. Thank you very much for the interesting talking, Carmen. It's quite impressive to see this behavior of the macrophages. They are always unpredictable. But well, perhaps you mentioned, but I didn't catch it. So in terms of the phenotype of the cells, if we think about the rounded ones and the stellate, yeah, perhaps it's easy to think that those which are activated will going to be the stellate ones. However, looking at your response, then both of them seems to be activated, and of course, since they are phagocyting and providing a response, that perhaps out of the TNF alpha, the response is not zero in any of those types. So, what exactly makes this difference in shape?
Yeah, this is a very great question. I think the shape is the parameter we use it to differentiate at the beginning these two cell types. And if I understand correct the question, what is the real function, because the shape is the form that we have to distinguish for the moment, but not if it's not that that the shape is important or making the difference in the function, because I think the stellate macrophages, that they can extend this protrusion to cap antigens, and I think that the rounded macrophages, they can do it as in mammals, that they can stand next to the epithelium and catch the antigen through that is going through the epithelium. So the shape there is not making the difference, and then for processing, shape is not important. Both are doing that, I think. And what we are analyzing now is what happens with antigen presentation to T cells, and maybe in this situation, yeah, it can make some difference. I don't know. Really, I'm very curious to see what happens, because as you see in the videos, the size and shape is real very different between these two populations. So yeah.
Yeah, definitely. The question was in that sense. So then both of them are functionally active and whatever, but those rounded ones resemble more a monocyte rather than a macrophage, a real macrophage. So then this was the question. And then I don't know if it's my view, but in your videos, it seems that at some point there could be some interaction between both types.
Yes, we have seen interaction between stellate and rounded macrophages, and we also have seen interaction between each macrophages population and lymphocytes and LCK positive T cells, but we need to have a way to have several colors to stain lymphocytes in a way, and macrophages, and of course the antigen, to see if it's really an interaction or just that they touch touch and go between the cells. So yeah.
Yeah, quite interesting. And just the last question, one more. Can I go through, Tiago?
See,
Of course,
please,
of course.
Yes, thanks. Yeah,
please.
so then you mentioned that perhaps the other cells that which are interacting are dendritic cells. What about some other cells, like it could be the Tregs or innate lymphoid
No,
cells?
uh, in mammals, goblet cells are the main cells that pass through antigens to immune cells. So it could be that this is also happens in zebrafish, but I think it should be another cell type too, because it's not that we are just quantifying those that are catching in this moment the antigen, but rather those that have this fluorescence from ova, and we have also do it the same experiment with the diet, so with different antigens at the same time, and we have the same results. So if this is the case, that globet cells are passing the antigens, immune cells should present it to T cells. So it could be, maybe we are thinking in this possibility also, that the time that the period of time that we choose for quantifying the cells is not enough to see more cells fluorescently labeled, and this is why we just see 40-50% of the cells positive for ova.
Well, quite a bunch of things to analyze and to try to determine. Perhaps you have there some primitive kind of M cells or something like that.
Yes.
Thank you very much, Carmen.
No.
Thank you, George. There's another question from the audience. It's Leonardo Oliveira asking, which food have you used to induce the inflammatory response?
We use soybean meal and fish meal. These are diets that are regularly used to feed fish in aquaculture. So we induce inflammation with a soybean meal based diet.
In terms of an ontogeny, these different macrophages, they have there is only one source of macrophages in zebrafish.
This is also a very good question, Tiago, because there it is reported that we have these M1 and M2 macrophages, so pro-inflammatory and more resolutive macrophages. But in terms of gene expression or gene marker, the main marker for the different types of macrophages that are used in mammals, they are not present in fish genome. So and this is not happens for dendritic cells. This is dendritic cells, lymphocytes, neutrophils. They have more or less the same marker. But for macrophages, is very strange, if we can say, because the main markers are not present.
But these ones are from hematopoietic compartment?
Yes. No, hematopoiesis is very conserved. We have all the waves, and we have this primitive and definitive macrophages. And in terms of function, they are also very conserved. But it's just that the that the markers that are used in mammals to identify the different populations in the gut, at least, are not present in the genome of the.
And another point, is there any way to deplete, or is
Yes,
this?
we are now using two strategies to deplete macrophages. We have we are trying crispants. These are doing these mutants in F0, so not real mutant, but with the CRISPR-Cas9 system. And we have also this the same system with nitroreductase that you can use it in mammals with a promoter that induce the expression of this enzyme in a specific population and deplete deplete one population and not the other. And these are both things that we are trying now.
I think that it's pretty much accurate, the strategy, the second strategy with the nitroreductase, because in this case, the cell ablation, it's very specific, because if you deplete all the macrophages and then you're working on the developmental stages, it's quite well understood that they will want to be required for some other functions that perhaps if the animal is defective, then by the end you will not going to have a clear response.
Yeah, I totally agree. It's easier with the CRISPR-Cas9 system, but yes, we will deplete it in a very long period and a lot of cells, and maybe not only macrophages will be affected, maybe other cell types. So just the ablation, but for this we need the construct also. Yeah.
Yes. You can start with a classical morpholino against the SPU1, and then that's a good try.
We can go to Jorge lab where everything works. So
welcome anytime, Carmen. You know that. Yes.
So yeah, I think you can finish. We don't have any further questions from the audience and Georgie and I. So I'd like to thank you, Carmen, again for this excellent talk. And I appreciate, and of course the organizer as well. And I hope you keep watching the symposium. And thank you very much again.
Many thanks again for the invitation. It's a great pleasure. See you. Bye. Thanks.
Bye. Thank you.
Bye.
Thank you, Carmen. Pleasure. Bye-bye.
So thank you for listening to this perfect presentation from Professor Carmen. And we'll be back at 3:30 Brazilian time for our next guest. See you soon, everybody. We'll be back. And now with our video from our sponsors.
O Instituto de Ciências Biomédicas da Universidade de São Paulo é uma referência nacional e internacional de qualidade no ensino, na pesquisa e nas atividades de cultura e extensão. Fundado em 1969, o ICB está localizado na cidade universitária, em São Paulo, com instalações em 8 prédios. Possui ainda uma unidade na cidade de Montenegro, em Rondônia, e um posto avançado em Cruzeiro do Sul, no Acre.
O ICB está estruturado em 7 departamentos: anatomia, biologia celular e do desenvolvimento, farmacologia, fisiologia e biofísica, imunologia, microbiologia e parasitologia, os quais contemplam as principais áreas das ciências biomédicas. Nosso ambiente é multicultural e amplamente democrático. Aqui no ICB, valorizamos a conduta ética, respeitamos a diversidade, incentivamos a consciência crítica e capacidade criativa dos nossos alunos, funcionários e professores.
Em meio a esse universo multidisciplinar, o ICB completa seus 50 anos com uma excelência consolidada e busca formar cada vez mais profissionais que produzam conhecimento e inovação, de modo a contribuir para o desenvolvimento da nossa sociedade. Seja você também parte do nosso Instituto.
Saúde, direito de todos e dever do Estado. Direito à moradia, ao emprego, à educação, a um meio ambiente saudável, à água potável, ao lazer, à cultura. Direito à voz e à paz. Respeito à diversidade. Saúde para a Fundação Oswaldo Cruz abriga todas essas dimensões. É assim desde 1900. Uma visão integrada da saúde. Na pesquisa, ensino, assistência, informação, comunicação, memória, na promoção da saúde, na inovação, onde permanentemente se pensam respostas para as necessidades da sociedade, onde se faz ciência em defesa da vida. Você pode até não saber, mas carrega a Fiocruz dentro de você. Desenvolvemos e fabricamos vacinas, medicamentos, biofármacos, testes para diagnóstico. Formamos pessoas do nível médio à pós-graduação. Realizamos pesquisas para superar ameaças à saúde, para diminuir riscos ambientais, para prevenir doenças e agravos. Pesquisas que beneficiam crianças, adultos, idosos. Uma instituição pública e estratégica de Estado, integrante do Sistema Único de Saúde, com uma rica história de contribuições à sociedade. Presente de ponta a ponta no Brasil, onde cada trabalhador é um elo forte e ativo. Nela, ciência e saúde cumprem uma função social para o país e o mundo. Pés fincados na tradição, olhos voltados para o futuro. Somos patrimônio da ciência e da saúde, da humanidade, do povo brasileiro.
Situada no estado do Rio Grande do Norte, no Nordeste brasileiro, a Universidade Federal do Rio Grande do Norte está entre as maiores universidades da região. Fundada em 25 de junho de 1958, possui uma área construída de aproximadamente 300 mil metros quadrados, que compõe os 5 campi distribuídos por todo o estado. Tem hoje mais de 40 mil estudantes divididos em mais de 110 cursos de graduação, mais de 90 cursos de especialização, incluindo as residências, 80 cursos de mestrado e 40 de doutorado. Conta com mais de 2.300 professores, dos quais mais de 1.500 com doutorado. São mais de 1.900 projetos de pesquisas desenvolvidos pelos mais de 340 grupos de pesquisa cadastrados no CNPq. mais de 700 projetos de extensão e 4 programas permanentes, além de mais de 3.000 servidores técnicos administrativos. Como uma universidade de ampla atuação, as atividades de ensino, pesquisa e extensão se estendem a todas as áreas: ciências humanas, ciências sociais, ciências exatas e da terra, ciências biológicas, ciências da saúde, tecnologias e artes.
Entre as áreas de destaque da UFRN, podemos citar o Instituto do Cérebro, que realiza pesquisas na área de neuroengenharia para o tratamento de doenças vasculares, epilepsia e outros distúrbios neurais. O Instituto Internacional de Física, que promove a troca de conhecimento científico com a comunidade internacional em áreas estratégicas. O Núcleo de Petróleo e Energias Renováveis, que por meio de parceria com a Petrobras, é considerado um dos maiores polos de pesquisa na área, formando mais de 1.000 profissionais em níveis de graduação e pós-graduação. O Instituto de Medicina Tropical, que tem por objetivo o estudo de doenças infecciosas com maior incidência em continentes de clima tropical, como América do Sul, África e Ásia. O Instituto Metrópole Digital, que atua na formação técnica e acadêmica por meio da inclusão social e digital de jovens, desde o ensino básico até a pós-graduação. A Escola de Música da UFRN, que tem por objetivo educar, produzir, disseminar conhecimento e fazer música como forma de contribuir para o desenvolvimento humano, promover a justiça social, a democracia e a cidadania. Nossa escola acolhe mais de 20 grupos e a Orquestra Sinfônica da UFRN, uma das únicas orquestras universitárias do país. O NUPLAN, Núcleo de Pesquisa de Medicamentos e Alimentos, um laboratório oficial destinado à produção de medicamentos para o Sistema Público de Saúde brasileiro. Entre suas atividades, o NUPLAN também oferece suporte acadêmico para a graduação e a pós-graduação, promovendo o ensino, a pesquisa, a extensão e a inovação tecnológica. O Ágora, Instituto de Línguas, Literaturas e Culturas Estrangeiras Modernas, que tem como principal objetivo oferecer cursos de língua estrangeira para estudantes, técnicos administrativos e professores da UFRN, bem como testes de proficiência para possibilitar a mobilidade internacional na universidade. O Ágora também recebe estudantes e pesquisadores das instituições parceiras da UFRN e de outros países, por meio do curso gratuito de português para estrangeiros.
Na extensão, o programa de maior abrangência é o Trilhas Potiguares, que leva o conhecimento de professores e alunos aos municípios do interior do Rio Grande do Norte, em ações sociais desenvolvidas em parceria com as prefeituras municipais. Com mais de 20 anos de atuação, o sucesso desse programa já o levou também ao exterior, com a ida de nossas equipes a Moçambique, atuando com as equipes locais. Outra ação de relevo da instituição é a Semana de Ciência, Tecnologia e Cultura da Universidade Federal do Rio Grande do Norte, maior evento de atividades científicas, tecnológicas e culturais do Estado e um dos maiores palcos para a interface da UFRN com a sociedade. Em sua estrutura, a UFRN também dispõe de 3 hospitais universitários, 2 deles na capital e 1 no interior, 22 bibliotecas, 3 museus, canal de TV digital, rádio, agência de comunicação, editora, núcleo de arte e cultura, núcleo de educação infantil, residência e restaurante universitário.
polo esportivo, disponibiliza campos de futebol e de treinamento oficiais, ginásios, piscinas olímpica e semiolímpicas. Assim, a UFRN busca a excelência e a inovação acadêmica por meio da formação de profissionais capacitados e ações educacionais comprometidas com a produção de conhecimento articulado com a integração social e comunitária, desenvolvendo soluções para os problemas regionais, nacionais e internacionais.
I'm the founder of Aquaneering. For the last 20 years, Aquaneering's built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community, and we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us, because for Alesco, research is for life.
Welcome everyone to FST's presentation. We appreciate you taking the time today and look forward to helping you find the right tools for the right application and your endless discoveries and research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.
My name is Mark Francis. I'm the founder of Aquaneering. For the last 20 years, Aquaneering has built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community, and we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us, because for Alesco, research is for life.
Welcome everyone to FST's presentation. We appreciate you taking the time today and look forward to helping you find the right tools for the right application in your endless discoveries and research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.
Hello, everyone. Welcome back. So we're going now for our today's fourth presentation. Dr. Matt Parker, who is a behavioral neuroscientist and psychopharmacologist, and he's also a professor at the University of Surrey. He leads a team that studies the biology of neuropsychiatric, neurodevelopmental, and neurodegenerative disorders, primarily using zebrafish as a model. His group also translates their findings to humans by having an active clinical research program with Portsmouth Hospitals University, NHS Trust. He has published nearly 100 peer-reviewed articles, chapters, and scholarly reviews. And he's here to present some of his findings and interesting discussions about neurosciences. So don't hesitate to participate, post your questions, and we'll be back for a discussion later. Thank you, Professor, for accepting our invitation.
Thank you very much. Can I just check that you can hear me? Yeah. Okay. So thanks very much for coming along to this talk. I'm going to be talking to you about some of the work that my group has been doing over the past 10 years or so, looking at developing adult zebrafish in particular as a model for neuropsychiatric and neurodevelopmental disorders. I should say that I've just literally moved to the University of Surrey today. Until today, I was working at the University of Portsmouth. So that's possibly what was on the bio that was online. But as of today, I'm working at the University of Surrey. So really the main motivation for our work is to try to develop precision psychiatry. Now, what I mean by precision psychiatry is that the kind of traditional concepts or diagnostic criteria within psychiatry tend to be fairly limited in terms of really understanding the biological basis of disorders. They're very convenient for clinicians in terms of pulling together a group of symptoms in order to understand, groups of people, how they may respond to particular treatments. But they do lack a precision, which, in my opinion, and in the opinion of many people who study biological psychiatry, really this doesn't tell us a lot about the underlying causes of the disorders, and indeed doesn't really help us towards treatments. And in fact, that lack of understanding about the fundamental biology, which potentially has been hampered by these kind of constructs within psychiatry, within clinical psychiatry, means that really the treatments that we have not moved beyond really the treatments that we've had over the past 50 years or so. Now, it's not to say that there aren't any new treatments, because of course there are, but they still lack the ability to treat all the patients and, to cure these disorders. So in fact, they tend to be quite palliative in nature. In other words, they treat the symptoms, but not the underlying cause. And we argue, as do many others, that if we're able to understand the fundamental biology underlying psychiatric conditions, then we might be in a better position to develop more precise treatments. Now, this may mean not just looking at constructs and psychiatric disorders as collections of symptoms, but instead focusing on individual differences at the patient level that allow us to target particular biological constructs that we may be able to design effective treatments for. Now, one of these biological constructs, if you like, that we're particularly interested in is impulsivity. Now, impulsivity, for those of you that don't know, is essentially a quite a broad range of behaviors that really talks about somebody's ability to withhold a response. So in other words, their ability to kind of hold back or to control their behavior before acting. And impulsivity is what we call a transdiagnostic. So in other words, it goes across a number of different diagnoses, neurocognitive phenotype. In other words, there are several diagnoses, several of these clinical psychiatric diagnoses, of which impulsivity is one of the core symptoms. And in fact, it's the quite a strong, in terms of the amount of variance predicted, phenotype for a number of neurodevelopmental and neuropsychiatric disorders. And these include autism, for example, and ADHD, but also things like schizophrenia and substance abuse as well. So it's quite a broad range of both neurodevelopmental and neuropsychiatric disorders. Now, just as an example of impulsivity in the context of substance abuse or drug addiction, I want to show you why I think that impulsivity is useful to study in terms of the underlying biology. Now, this is a figure from a paper in Neuron about 10 years ago or so now by Jeff Daly and colleagues at Cambridge. And what they were looking at was individuals that were substance abusers, so regular drug users. And what they were looking at was the extent to which impulsivity as a neurocognitive phenotype, so as a personality trait, was related to drug abuse. And what was quite interesting about their approach was that they didn't just look at drug users, they also looked at their first degree relatives. So they looked at their siblings and they compared these to the general population. And what you'll notice was that when you look at sensation seeking, which is, in other words, maybe doing kind of crazy behaviors, so jumping from an aeroplane with a parachute, or doing very high novelty seeking activities, so people that are really like to be very overexcited. They found that this was higher in drug users, but there was no difference between the general population and the drug users' siblings, suggesting that this was something specific to drug users, potentially something that comes after the drug use has taken place, or some kind of gene environment interaction, for example. However, with impulsivity, what was really interesting about this subtype impulsivity was that not only did drug users show higher levels of impulsivity than the general population, their siblings also showed significantly higher levels of impulsivity to the general population. So bear in mind, these are non-drug user siblings. So what this suggests really strongly is that impulsivity as an underlying trait must have some kind of genetic basis. Now, stronger than this was some work that was carried out by Bellin and colleagues, again from Cambridge Neuroscience. This was a little bit earlier than that, about 2009. Now, what they found was that they trained rats on a task of impulsivity. So in other words, a task where the rats had to withhold a response in order to get access to a food reward. I'm going to talk a little bit more about the protocol that they use, because we use the same protocol in our zebrafish. So I'll explain the protocol a bit later. So maybe keep this at the back of your mind as the same work that we do with the zebrafish, and I'll explain it later. What they found was that rats that were categorized as high impulsivity, so in other words, rats that showed a high level of impulsive responses as compared to those that showed low impulsive responses, were much more likely to show compulsive cocaine use. Now, what this was that they gave the rats essentially access to cocaine, but it was contingent on getting an electric shock. So once the rats had learnt that they were able to get cocaine by pressing a lever, they then put that response for cocaine on an interval schedule, which meant every certain number of presses they might get cocaine, but they might also get an electric shock. And what you'll notice was that the low impulsivity rats, they stopped responding. They said, okay, I like cocaine, but I don't really want to get an electric shock. Whereas the high impulsive rats, they showed very high levels of responding even when they were getting the electric shock. And what was also really interesting is that they measured the impulsivity of these rats, and then they plotted that as a function of their premature responses. So in other words, their impulsivity. And what they found, which was really interesting, was that the animals that were high in impulsivity were also high in compulsive responding. So in other words, there seemed to be a strong positive correlation between the likelihood that the animals that were high impulsive would also be high compulsive drug seekers. Now, this was quite intriguing because it suggested not only that impulsivity is important in terms of a personality trait that predicts addiction, but also that it leads probably in a causal way to compulsive drug seeking. Because bear in mind, this correlation was carried out before the animals had had any access to drugs. So it's a causal factor potentially. Now, not only was this work showing that in terms, sorry, I'm just trying to remove that from my screen. There's something blocking my screen and it's making it hard to see. Not only did they find that there was a increase in compulsive drug seeking, they also found that the high impulsive animals were more likely to show punishment-induced relapse. So in other words, when you essentially give an animal access to drugs and then you put them into extinction, so in other words, you no longer have a contingency, so you press the button and they're not able to get access to the drug anymore. When you put them into that extinction, then normally the animals will not reinstate their drug seeking if you put them into extinction. But what they found was that the animals that were high impulsive, when they had to induce punishment, so in other words, when they did an electric shock, these animals were much more likely to reinstate their drug taking. Okay, so it made no difference in terms of their extinction, but as soon as they had extinguished the responses and they gave them a punishment, the high impulsive rats started seeking the drug again. So not only does impulsivity seem to predict compulsive drug seeking, it also seems to predict relapse. So in other words, it's got a lot of the different underlying factors that we see in substance abuse and in patients that show substance abuse. So for these reasons, they also show higher, I should mention the high impulsive also show higher rates of self-administration of drugs as well. So what's quite interesting at this point is to note that impulsivity, although it's not necessarily directly related to the drug taking itself, does appear to make animals and humans and their siblings at higher risk of compulsive drug seeking and relapse, and indeed also potentially of the rate of administration, so how much of the drug they take at any one time. So how do we measure impulsivity? Well, in humans, there are several ways of measuring impulsivity that typically involve either questionnaires, psychometric scales, or behavioral tasks. And we use these in my lab quite a lot. But today we're talking about zebrafish. And so we want to talk about some of the ways that we measure impulsivity in animals. Now, as I mentioned at the beginning, impulsivity is the animal's or the person's ability to withhold a response. So in other words, when it is not appropriate to respond, they need to be able to override the wish to respond or the drive, the motivation to respond, and withhold that response. So the way this can be measured in the lab is by carrying out a task where the animal has to pay attention to a series of cues. And when it sees one of those cues, it responds. And if it responds correctly, then it gets rewarded. So in this protocol here, this is a protocol called the 5-choice serial reaction time task. So you can see that the animal has 5 holes in a 5-hole box. And what will happen is that the box is in darkness. And after a delay of a certain number of seconds, one of these apertures, one of the holes in the box will light up. And when that hole lights up, the animal has to poke its nose into that hole. And if it gets the correct hole, then it will give a magazine light at the opposite end will be illuminated, and the animal can turn around and then go and receive a food reward. Now, the way that impulsivity can be measured within the context of this protocol is that you can vary the delay before this aperture light comes on. So before one of these lights comes on, it stays dark for a certain period of time, and then the light comes on. And what happens is that the high impulsive animals, the animals that find it hard to withhold a response, when you put them under continuous performance schedules, so in other words, they're having to do this task for a long period of time and they're getting rewarded for doing it over a long period of time, many trials over a long period of time, some animals will show high rates of premature responding. So in other words, they'll respond by poking their nose into one of these apertures before the light has come on. Now, that premature responding is a way of operationally defining impulsive behavior, because the animals are unable or unwilling, but probably unable to withhold that response, despite the fact that they know that it will, if they poke their hole in prematurely, it will stop the task and they won't get rewarded. So there is a penalty, a cost associated with that response. And that's really important in the context of impulsivity. So despite the animal being aware that they are being punished for making that response, they still make the response and hence are showing impulsivity. Okay, so that gives you an overview of impulsivity and how it relates to neuropsychiatric and neurodevelopmental disorders, but also how we might measure it in an animal model. So I want to talk to you now a little bit about how we've measured this in fish. Now, I don't need to go into lots of detail about how fabulous fish are. This is a zebrafish webinar, so everybody here knows that zebrafish are a great model system. Again, I don't need to go into lots of detail about the fact that we know a lot about the zebrafish genome. It's been fully sequenced, and there are going to be some fantastic talks in this webinar about genome sequencing and so on. They also have high fecundity, so they lay many eggs, relatively low cost. Obviously, they're a vertebrate, they have a fast life cycle, and importantly for our work in particular, they are robust to mutations. In other words, you can knock out genes in zebrafish that you perhaps are more difficult to knock out in mice, for example, and still get survival of the animals, potentially because of genome duplication events and so on, but nevertheless, they are particularly robust to mutations. Okay, so how do we measure impulsivity in the zebrafish? So I've already given you a clue because I told you about the 5-choice serial reaction time task. So let's have a look at that in a little bit more detail. So this is a figure from a paper. Actually, it's not from that paper that I put down there. It's from a different paper by Barbara Fontana, one of my old PhD students. But we first described this task in zebrafish back in 2012 in a paper in behavioral brain research. So that was back when I worked in Caroline Brennan's lab at Queen Mary in London. So essentially, the 5-choice serial reaction time task in a fish is no different to the 5-choice serial reaction time task in a rat that I have already described to you, except for a couple of key features. So the first feature in a fish is that the size of the arena compared to the size of the animal is somewhat larger. We tried during the development of the task to have it in a much smaller arena, but fish get very stressed in a very small enclosed arena. And so, rats are relatively comfortable in those types of arenas. So we had a slightly larger arena for the fish, so about 20 centimeters long. We also train the animals on a slightly different schedule to the way that they train rats, in the sense that with rats, often what they're trying to look for is attentional responses as well. We have never designed this task in particular to look at attention because the fish are kind of swimming around and not sitting in one position, performing orienting responses. So we set up our task by trial starting, by the fish initiating a trial by swimming over this initiator light, and then there would be a delay, and then one of the stimulus lights would come on. So we train the fish over a number of days. This is just an overview. Often it takes longer than this, but roughly sort of 5 to 6 weeks in order to get them trained up to be ready to, and that's every day of training to get them ready to perform this task. And similar to the rodents, what we were looking at was the fish's ability to withhold a response before that stimulus light came on. Okay, so once they trigger the initiator light, the fish then has to wait, and then one of the stimulus lights will come on. The fish swims into that area. It's then, if it gets it correct, it then gets reinforced by swimming back to the food hopper where it will be reinforced, and then it can start the next trial when it's ready. So by swimming over the initiator light, that will then start the subsequent trial, and then they carry on. Okay. So this is just an overview of the task in that I've just described. So some of our early work in this, what we were interested in was sort of baseline levels of premature responding, but also how they responded to low levels of a psychostimulant, because as you probably know, psychostimulant drugs, so noradrenergic or dopaminergic reuptake inhibitors, such as amphetamine, or amphetamine substrates like methylphenidate and so on, are used as a treatment for attention deficit disorder and various things like that. So in our early studies, we used amphetamine, a very low concentration of amphetamine, in order to try to reduce premature responding, and we found that a low concentration of amphetamine reduced premature responding, but had no impact on the other aspects of response, suggesting that fish were a suitable model species to carry out this kind of work. So this was the early work that we did on impulsivity, was not done automated. Okay, so the equipment that we used was manual, so we were having to use like paper and cardboard and plastic and stuff to lift things up and trying to reinforce the fish manually, which obviously wasn't very good and did not allow us to carry the work out high throughput. So we developed a system which allowed us to automate the impulsivity measures. So we published this a couple of years or a year or so after the original paper, and this is a video of a fish carrying out the 5-choice test. So you can see here, this fish has just been rewarded at this end. It then swims up to the other end and it sees the light. It swims into the correct arena, and then it goes down to the other end and gets fed. You may notice in the center of the tank here, so this is a premature response. So this time it didn't wait. So unfortunately, it returns back to the beginning of the task, and it has to start the task again. So that impulsive response was punished. So we trained the fish on essentially a random interval schedule of that pre-stimulus interval, and that allowed us to phenotype the fish. In other words, to see phenotypically which fish were more impulsive than others. Now, the data for this were really nice. So we looked at the number of anticipatory responses during training, and as you'll notice, those kind of premature impulsive responses reduced as a function of training, so up to about day 20. And the correct responses, so in other words, how often the animals made a correct choice, as opposed to incorrect. So in other words, that incorporates only correct and incorrect trials. They got up to around 50%. So of course, it's well above chance, which was around 20%, but it was still fairly low. But it was okay, and we were relatively happy with that. Now, one of the things that we really wanted to look at with this was whether there was any way of kind of increasing the correct responses and make it a little bit more like the rodent tasks. And we thought that might have helped us in terms of trying to understand more about the genetics underlying impulse control, if we were able to sort of carry out more tasks, make it a bit more high throughput, but also have a better level of responding so that it was more akin to the rodents. Because obviously we have to keep the people that work with rodents happy, otherwise they'll say it's a different task, it's measuring something different. So we work with a company called Xantix, which some of you may have come across before. So Xantix make a number of different inserts for their recording tanks, and they make a 5-choice serial reaction time task, which in fact was based on our work anyway. So they custom made it for us, but it works really well in that context. And so this allowed us to have everything on a slightly smaller scale, but also have a lot more precise automation for the task. So this is a video of the same task, but with the Xantix unit. So you can see it's slightly smaller. The fish swims around and one of the lights comes on. Excuse me while I just have a drink. And so the fish clicks the initiator light. And once it's clicked the initiator light, one of the other lights comes on, and then it swims back and it gets fed. So this way of doing it made it, allowed us to do it faster. And actually, what was really good about this was that it actually increased the number of correct responses. So in other words, as I mentioned before, we wanted to try and get the correct responses around above 50%. And in fact, with the Xantix unit, we managed to get the amount of correct responses right up to 80%, which is what you see in rats, and the number of emissions, so trials where the animals didn't respond, which I should have said actually with the older system was much higher. The emissions went right down to much lower levels. So a much better way of measuring it. We also then published a paper in psychopharmacology where we looked at the effects of atomoxetine, which was a noradrenergic reuptake inhibitor, on the 5-choice test within zebrafish. And in fact, we found that the anticipatory response is reduced in a dose-dependent manner using atomoxetine. So, excuse me. So collectively, what these data show us is that not only are zebrafish able to carry out a task of impulse control in a similar manner to what we would see in rodents, they also respond to noradrenergic drugs in a similar way to rodents, and indeed in a similar way to humans. So all of this work really allowed us to be confident that we were measuring impulse control in zebrafish, but also that impulse control was probably under the same neural circuits, so the ascending noradrenergic pathways, as we see in rodents and in humans. Okay, so on the basis of this impulsivity task, how could we use that to understand more about the genetic basis of impulsivity? So I just want to go through a couple of examples of how we've used that task and similar tasks to try to understand a little bit more about the genetics of impulsivity. And this is an area that we're currently working on and is being expanded all the time. So, you know, this is something that we're very much looking towards in the future. So again, I'll tell you some work that we've been doing recently, and then some of the work that we're planning to do in the future. So there are lots of genes that have been identified, for example, through GWAS studies, genome-wide association studies, and linkage studies, and so on, that have been related to impulse control disorders. Not just impulse control disorders, but also sort of general externalizing conditions. So disorders that are related to kind of impulsive and compulsive behavior, some of which we've already mentioned, but several others. So one such gene of interest is, one such disorder, if you like, or one such condition of interest is obviously nicotine dependence. And there's been lots of, because obviously we know that smoking is related to kind of impulse control, habit formation, and so on. And people that are dependent on nicotine clearly find it very hard to stop smoking. And again, that's linked, that sort of compulsive nature of smoking is linked to other impulse control disorders. Now, one particular gene of interest that has been related to nicotine dependence in particular is the beta-3 nicotinic receptor. So the alpha-6 beta-3 nicotinic receptor has been strongly linked to nicotine dependence in GWAS studies. What was quite interesting was we found somewhat serendipitously, I should mention, when we were looking at something else, that a particular strain of zebrafish that have been used quite a lot for looking at muscle development, the ache fish, which are they're heterozygous for a knockout in acetylcholinesterase. So they actually produce a slightly lower amount of acetylcholinesterase. The homozygous animals are non-viable, but they're heterozygous, so it's a sort of gene dose effect. So they have a reduction, these animals, in the beta-3 nicotinic receptor mRNA expression. I should say this is published in Frontiers in Behavioral Neuroscience a few years ago. So as you can see, they have an increase, I beg your pardon, the knockouts in the beta-3 nicotinic receptor. And so what we did was, based on the hypothesis that these animals had lower beta-3 nicotinic receptors, we thought that potentially they may have differences in impulse control. I should have mentioned, actually, sorry, let me just go back here. One of the reasons we think this might be to do with dependence is because that alpha-6 beta-3 sort of genetic, like in the GWAS, was not related to heaviness of smoking. It was only related to dependence, right? So it's not necessarily related to the nicotine or the cigarettes itself. It's potentially related to the dependence. I should have mentioned that. What we did was we screened these animals for impulsivity, and sure enough, the animals that had higher levels of that beta-3 nicotinic receptor also showed lower levels of impulsivity. So on this graph here, you can see anticipatory responses in the wild types and the knockout animals. And again, you can see on average when we tried so this was during training, and you could see that after training, once they were established, the knockout animals showed significantly lower, about half the number of anticipatory responses, so premature responses, than the knockouts. So potentially, this was quite interesting. So we actually looked at a number of different nicotinic-related and cholinergic drugs, and although those cholinergic drugs increased the correct responses, they had no significant impact on anticipatory responses. And what that suggested was that it wasn't just to do with higher levels of acetylcholine that was causing this premature responding. It must have been something to do with the sort of developmental effects of having lower levels of acetylcholinesterase during responding, or higher levels of acetylcholine. So this was quite nice because it was really the first demonstration actually in the literature of a link between cholinergic signaling and impulse control. And this is something that we've been following up recently. So in terms of other genes that have been involved with impulse control disorders, one particular gene of interest that's been involved, that's been related to attention deficit hyperactivity disorder susceptibility, but also to substance abuse and several other impulse control disorders, is the gene that encodes the adhesion G protein coupled receptor L3, or ADGRL3 for short. It's also known as latrophilin 3, so some people may have come across that before. Now, that's been strongly associated with ADHD in a number of different independent samples. So it's not just one finding. In genetic linkage studies, that particular gene has been linked strongly to ADHD. As I said before, it's also very strongly related to substance abuse disorders independently of ADHD, suggesting not only is that it's important, but also that it's probably important for impulse control, because substance abuse and ADHD, although they're similar, again, this gene independently predicts susceptibility to both. So it must be involved with some underlying factor of both of those disorders. So despite the fact that we know this, and we found that in genetic linkage studies, we don't really understand the sort of underlying pathogenic mechanisms of this ADGRL3 difference, if you like, variation in impulse control disorders. So about 10 years ago or so, Will Norton's group knocked out up at Leicester, although he was working in Laure Balaquif's lab at the time in Paris, knocked out, used a morpholino to knock down ADGRL 3.1, and looked at the resultant phenotype, and they found that it increased hyperactivity. They also did some work on impulsivity, but not using adult impulsivity. They were just looking at kind of burst swimming, so kind of impulsive like swimming patterns in the larvae. And what they found was that when you knock this gene down using a morpholino, it increased hyperactivity, but that hyperactivity phenotype was rescued by atomoxetine and also by methylphenidate. They published that in Molecular Psychiatry about 10 years ago. So we've recently been working with Carl Carlson's group in Iceland. And Carl's group have been looking at drug development studies for ADHD, and they have also been looking at the ADGRL 3.1, and they have used it in terms of looking at, as I say, drug development studies. So what's really interesting about this, so now we've used a CRISPR to knock out ADGRL 3.1, and we have a homozygous knockout. And Carl's been using it for this work, and we've also been using it for some other work I'll talk to you about in a moment. So not only do we find that this ADGRL 3.1 shows high levels of hyperactivity as larvae, they also show disrupted sleep, and that's something that we know with people with ADHD is a particular problem. So we also know that the phenotypes that we see within the ADGRL 3.1 are rescued very effectively by atomoxetine. Okay, so again, that noradrenergic system, although not directly related to ADGRL 3.1 protein, appears to be very important. So again, that motivated us to look in more detail at impulsivity in these animals. So this is some fantastic work by Barbara Dotto Fontana, who was working in my lab, who is now working over in the States. And this was some work that Barbara did during her PhD, and also involved was Will Norton from the University of Leicester. So what we did was we took ADGRL 3.1 adult fish and we screened them on our 5-choice serial reaction time task. So you can see the data on these left two graphs here. So we found that the ADGRL 3.1 knockouts, so homozygous knockouts, we also did heterozygotes as well, but I've not shown the data here. Heterozygotes show the phenotype, but at a lower level, but the data is not shown here. So they show lower levels of accuracy. They also show much higher levels, so nearly double the average level of premature responses. And what's really interesting about it is that that's completely rescued by atomoxetine. Okay, so the accuracy is improved, suggesting that they show better levels of concentration, perhaps, but also their anticipatory responses, so their impulsive responses, are completely rescued by atomoxetine. So they go right down to wild type levels. We also looked at an additional measure of impulsivity, which is a novel object test. Now, I don't want to kind of get into an argument about that, because some people will say that it's not impulsivity. I tend to agree. It's probably more boldness or novelty seeking, but it's kind of like a bit, you might think it's similar to the human sensation seeking that we talked about before. Interestingly, we also found that the ADGRL 3.1 knockout animals show high levels of interest in the novel object, and that again is rescued, if you like, that phenotype by atomoxetine. So what we did then was because ADGRL 3.1 itself it's difficult to understand what the mechanisms are by which that particular protein is impacting on impulsivity. What we did was we carried out RNA-seq, so we looked at transcriptomics of the animals that had the ADGRL 3.1 knockout versus wild types. And what we found was several differentially expressed genes. And what was nice about this, I'm not going to go into the details of them now, this will be published fairly soon, we're just working on the paper. We found several genes that actually come up in ADHD GWAS studies, suggesting that downstream of that, we may well have potential targets that haven't been considered before for ADHD medications and therapeutics, but also might help us understand a little bit more about the the kind of functional pathways that underlie impulse control disorders. Okay, so just to summarize, impulsivity is a core transdiagnostic endophenotype for several different disorders that we see in siblings and the people affected with the disorders, suggesting it has a strong kind of biological basis to it. Zebrafish show robust levels of impulsivity. Again, we don't know yet about the larval stage. I've kind of teased that a little bit with the work that Will Norton's group did with larvae many years ago, and that's something that we're looking at the moment, is whether you can predict adult impulsivity from larval behavior, but that's something that we're working on at the moment. And we've shown fairly strongly, in my opinion, that genetic lines with genes that are strongly related to impulse control disorders in humans show different rates, higher rates of impulsivity that we're able to treat with drugs that effectively treat impulsivity in humans. And this means that we may well be able to use fish for finding novel genes or novel targets that might be useful for treating impulse control disorders. So thank you very much for listening. It just remains for me to thank the people that I've worked with on this over the years. I mean, obviously they did all of the work, not me, so it's important that they're acknowledged, especially Barbara Fontana, who did a huge amount of that impulsivity work that I talked to you about, but also collaborators, especially Will Norton and Caroline Brennan, and all the technical staff, and also all the fantastic funders that we've been lucky enough to be able to extract money from over the years to carry out this work and for the continuing work. So yeah, that's the end of my talk, and I'd be very happy to take some questions from the audience.
So hello, Dr. Matt. Thank you for the perfect presentation. It was very, very interesting. And we have two questions from the audience. The first one is from Ana Luciari, and she's from the Federal University of Rio Grande do Norte here in Brazil. So her questions are: how is impulsivity presented along ontogeny in zebrafish? are younger animals more impulsive than older ones? Does it have any relationship with GABAergic system maturation?
Yeah, it's a great question, Anna. Thank you for that. So I probably answered the ontogeny one a little bit earlier. So that's something we're interested in at the moment. I mean, there's obvious benefits in terms of throughput, drug discovery and so on, if we were able to find larval phenotypes. In terms of the general kind of maturation process, we don't see big differences, in fact, between, for example, 3-month and 6-month-old animals. One of the problems is when they're younger, they don't eat as much, right? So they're not as big physically. So it's hard to compare, because when you're training them on one of these 5 choice, they have to eat a lot of food day after day after day after day, and the appetite maybe isn't as big in the smaller fish. So we don't see such good rates of responding. That the perfect time is around 6 months. We've not tried this in old animals, so maybe a year old, maybe 2 years old. Those of you that have seen me talk before will know that my lab also does a lot of work on memory, and we found very, very striking differences in old fish, so fish that are kind of 2 years old and older. They show cognitive decline. So I think it'd be really interesting to look at whether there is a nice ontogeny in terms of the sort of development of impulsivity, but it's not something we've looked at. But we are really interested in trying to figure this out in larvae, and this is something, in fact, that we're trying to do as we speak. So we've applied for some grant money to do that. But thank you. That's a really good question.
Yeah, very nice. The next question is from Tan from the University of Liège, Belgium. And he's saying, thank you, Dr. Parker, for your fantastic talk. And he'd like to ask if you have any recommendation for larval assays of impulsivity.
Yes,
for
I see a
example,
theme.
open field.
Yeah, I see a theme here. Yeah, no, again, everyone's thinking along the same lines as me, which I like. Yeah, I think we really do want to find. I'm not sure about open field test. I mean, it's possible, but it's hard to say whether that is impulsivity or not. I mean, if we find something that predicts impulsivity in older animals, then we could argue it's impulsivity, I guess. At the moment, we've not seen any sort of typical larval movement related endpoints that have predicted impulsivity. The only thing I would say is obviously hyperactivity seems to predict, but we think that not just we think, there's very good evidence that those two behavioral constructs are different pathways, right? So even though we see predictive validity of hyperactivity and impulsivity, I don't think they're the same mechanism. So yeah, it's a great question. It's something we're really interested in doing, and something that we're actively pursuing in the future.
Nice, very nice. Other people are saying congratulations for your talk, Dr. Parker. It was amazing and excellent talk.
Thank you for coming.
Does anyone in the audience have another question for Dr. Parker? I don't think so. I think it was only this.
Please do email me if you have any questions. Email me. I'm more than happy to go over any of this in the future, or give me a follow at PsychGenes on Twitter, and we talk about these kinds of things all the time.
I'm gonna follow you on Twitter.
There you go. No worries.
And so I think that's it for now. And PsychGenes. I'm gonna post the dots from Twitter here on the comments. So this the one in the screen now is the how to find him in Twitter, people. And so we're gonna be back at 4:30 for our next presentation. Thank you very, very much, Dr. Parker. It was lovely to have you.
No problem.
And now we are gonna present our sponsors video while we wait for our next guest.
Right.
So see you in a bit. Bye-bye.
Thanks. Bye.
My name is Mark Francis. I'm the founder of Aquaneering. For the last 20 years, Aquaneering's built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community, and we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us, because for Alesco, research is for life.
Welcome everyone to FST's presentation. We appreciate you taking the time today and look forward to helping you find the right tools for the right application in your endless discoveries and research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.
My name is Mark Francis. I'm the founder of Aquaneering. For the last 20 years, Aquaneering has built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community, and we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us, because for Alesco, research is for life.
Welcome everyone to FST's presentation. We appreciate you taking the time today and look forward to helping you find the right tools for the right application and your endless discoveries and research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps, and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.
Hello, everyone. Good afternoon. Let's start our next session. I will present the next speaker, Dr. Oscar Ruiz. He grew up in Brownsville, Texas, but he's originally from Tampico, Mexico. Despite this, he considers himself a native Texan. After high school, Oscar attended to the University of Texas at San Antonio and received his bachelor in biology and finished his university training with zero research experience. After the graduation, Oscar moved back to Mexico and taught HS biology for a year. During this time, Oscar also enrolled in a master program in food science and biotechnology at the University Autónoma de Tamaulipas, where he studied the chemical composition of various agave species that are used as food sources. After finishing his master's degree, Oscar moved back to Houston to work at Baylor's College of Medicine as a research technician in the lab of Dr. Monica Justice. During this time, Oscar worked on a genetic screen in mice looking for mutations that affect metabolism. After his time as a research technician, Oscar began his graduate studies at the University of Utah in the lab of Dr. Mike Metzstein, where he worked on the development of the larval Drosophila respiratory system. After he graduated, Oscar pursued his interest in host pathogen interactions by moving to Lisbon, Portugal, for his postdoctoral work in the laboratory of Dr. Luis Teixeira. There, his work they're largely focused on host microbiome interactions in Drosophila and specifically on the identification of Drosophila genes that are important for its immunity devices. In 2014, Oscar came to Texas to work at MD Anderson Cancer Center in the laboratory of Dr. George Eisenhofer, where his research focused on studying tissues homeostasis in epithelia and the opportunistic infections that can arise when epithelia are damaged. In White Laboratory, Oscar leverages his experience in imaging and zebrafish, as well as bioinformatics, to understand how endothelial dynamics are altered in disease settings, such as brain arteriovenous malformations. Oscar also teaches currently at Houston Baptist University and is actively involved in educational outreach and continuing education. Welcome, Dr. Oscar. It's a pleasure to have you here. I hope you can start now sharing your screen.
Thank you so much, Joao. I should be sharing now. Can you see it?
Not yet. Yeah, it's
there. We go.
It's okay now.
Perfect. Thank you so much for the introduction. That was a bit long, but I appreciate it. So yes, all my talk today is going to be about work that was done when I was a senior scientist in the laboratory of George Eisenhofer here at MD Anderson Cancer Center. And so, showing you a picture of this is a face of a zebrafish embryo. Our previous talk focused a lot on adult zebrafish. We're going to go back and just do embryos for my whole talk. And so, let me see if I can get this to work. For some reason, I can't get the slide. There we go. Okay, there we go. So in the Eisenhower lab, we're super interested in tissue homeostasis. And so if you ever look at a histology slide or a textbook, you'll see some sort of this kind of epithelial structure, right? Where you have some cells, not a lot's going on. But one of the really hidden things that get passed off when we look at a normal tissue or things like that is that there's actually a lot of things going on in that tissue. We think it's very stable and very slow, but there's actually a lot going on. And mainly, there's always cells that are being lost off the surface, and there's cells from the bottom that have to migrate up to seal those holes, right? And so we're interested in how these tissue cultures, how these tissues are able to control the amount of cell division and the amount of death to coordinate the overall numbers in epithelia. And now normally this happens, we don't really think about it, but if one day you go outside, you cut yourself, that leads to tissue damage, right? To where you lose a bit of that tissue. Now, for most of us that are okay or not suffering from other problems, our tissue is able to recover perfectly fine, and we get a nice sort of closing of that wound, right? This is normal wound recovery. Now, if we can't do that, we have a situation where we have impaired wound healing that allows for opportunistic infections that can now come in to that before area that was closed. Conversely, if the cells proliferate too much to close that wound, we end up with something like cancer, right? So cancer can be thought of as a situation where you have a wound, the cells begin to divide to close that wound, but then it never stops, and you end up with this cancer. And so we're really interested in how this happens. And so for that, we turn to the zebrafish, right? Obviously, this is a zebrafish webinar, so we're only talking about zebrafish. But since this is meant for undergraduates, I want to talk a little bit about why we do zebrafish, or some of us do zebrafish. The main thing is they're small, right? They're really small, they're easy to maintain, and they have a very short generation time, meaning that we can go from 0 to 3 months to breeder animals in 3 months. So that's really fast. Now, some of the other reasons we do it is because they develop fully externally, right? So we can watch whatever they're doing. Additionally, when they go through development, they go super fast with development. And then one of the things that I love the most, being a microscopist, is that they're transparent, right? So it makes it really easy to image right through them. And so you might be thinking, how do we go from an embryo, this one cell stage, to an actual fish? Well, it's actually, because we can film it, here's a movie of this happening exactly. We start with 2 cells, divide into 4 cells, and they go in through all these different cell divisions through the process of epibole, gastrulation, and you end up with a small fish, right, an embryo, in about 24 hours. So pretty soon here in this movie, you're going to see somewhere here the eye or the optic cup develop. At the very end, you're going to see that it's essentially a little fish. So here, if you want to look right around here, this is going to become the optic cup that will eventually lead to the eye, zebrafish eye, right? So this is about 24 hours of development. So development happens super fast, and we can watch it all happen outside mom. Now, here's a staging series, right? We go from one cell stage to, in 36, 48 hours, we have essentially a swimming fish, which is super interesting for us and allows us to do a lot of different things. And so I told you about epithelia, right? I'm interested in tissue. And so the fish have a really cool epidermis, right? They have this cells that cover their outside skin. And in this picture here, I'm showing a transgenic fish that's labeled all the outside epithelia with a transgene called keratin4. So keratin4 is, in this case, is hooked up to GFP, and so we're able to label the entire epithelia of the outside of the zebrafish. Now, the important thing about epithelial in the zebrafish is that during development, it's actually a bilayer, right? Which means that it's composed of 2 cell types, 2 cell layers. And if we zoom into one of these areas, we can see these 2 layers. So the top layer, which is called the periderm, is the layer that faces out to the outside world, right? Exposed to the environment. And then the layer right beneath it, which is we call basal cells, we can think of them as essentially the stem cells that are to divide and replenish any periderm cells that may be lost. And so just to show you that this is very similar, I use on the right here, I have a stain of keratin4, which is stains mainly differentiated keratinocytes, right? So we think the outside or periderm cells have gone through differentiation. And then right underneath it, that basal layer, I'm marking here with an antibody stain for P63, which is a known marker of stem cells, especially epithelial stem cells. So we see the stem cell layer, and above that, the differentiated layer. Now, just like I told before, right, we think of this sort of static thing, but it's actually very dynamic, right? The homeostasis, a lot of things are happening at the same time. And one of the main things is that these periderm cells, or the cells that are exposed to the outside, they're constantly facing insults, right? They're outside, they're getting bombarded with toxins, sun, anything you can think of, and these cells are being damaged. And so what happens when you have one of these cells being damaged, the neighboring tissue can actually sense that. And when they sense that, the neighboring cells begin to push. They use mechanical pushing to sort of pinch out that cell through a process called cell extrusion. Now, this is just a drawing or a schematic of that cell being pushed out, but this is what it really looks like. This is a scanning electron microscope picture, and the cell in magenta, the cell in the middle, is actually the one that's being extruded. It's the cell that was damaged. Those neighboring cells can sense that, and they're all using that force to push that cell out of the layer. So you don't want any damaged cells sticking around, right? That leads to bad things. So this is how the tissue deals with this. Now, if we look at this, that was an SEM, right? So that's a static movie. But what happens is actually really cool to watch the movies. This is a movie that one of our former postdocs, Yumna Adie, was able to take. And I want you to focus on this one cell, this big cell, right where the arrow is showing. And so they're going to see a series of contractions to that tissue. And every time they contract, that cell's going to get smaller and smaller and smaller, and eventually get pushed out of that again. So you see contractions, and at the end, it actually gets kicked out. So these contractive pulsations are super important in being able to push that cell out of that tissue. Now, this response, right, is super fast, right? You're able to push that cell out and close that gap. And so the importance of that is that you're able to maintain something called barrier function. Barrier function means that you want to keep everything that's outside, right? When you're pushing that cell out through extrusion, you don't want to have any gaps where things can come in. And normally this happens really well. When it doesn't happen super well is when you have a ton of damage, right? So if you have a ton of damage, that means that instead of pushing out one cell every 10 or 15 or 20 cells, you may be having to have a lot of cells damaged, and they all have to get pushed out at the same time. Now, what happens is that when that happens, you end up with a loss of barrier function. And that leads to things that are normally on the outside that normally couldn't get in because of that functional barrier function. Things like viruses, bacteria, or fungus now have access to the basal stem cells, right? So that cell layer that they couldn't get to before. And so this is something called opportunistic infections, right? Something where things that are normally on our skin can't do anything, but then when we have a cut or we can't regenerate our skin, then these things can take hold, right? And so we're really interested in that. And so if you think about when that happens, cancer treatment is really one of these things that causes widespread tissue damage, right? And so when you go and you get radiation treatment or chemotherapy, things like that, you're really blowing up all your skin, right? And that leads to opportunistic infections. And so if you think about this opportunistic infection in a setting of cancer treatment, one of the biggest things other than having cancer from cancer is getting something called mucositis or neutropenia, right? This is where mucositis, you lose that barrier function, and neutropenia, when you're killing your immune cells that are there are meant to go around and kill things that come in. And so these are two really important infections or really important side effects that come from the cancer treatment itself. And so if we can figure out how that works, maybe we can figure out a way to hopefully protect patients before they develop mucositis or neutropenia. And so we turned to the fish, right? We thought, well, how can we do this in fish? So we made we have a protocol called controlled ablation of scattered epithelial cells, or KSEC. Now, the important thing is that we have a transgene, in this case, a transgene controlled by GAL4 that's driving a UAS NTRM cherry. Now, that sounds like a lot, but the important thing is that if you look at the schematic here, that not every cell is pink, right? So it's mosaic. It's mosaic expression, meaning that the top paradigm, not every cell is being labeled. And so normally nothing happens. All you do is you have a subset of these cells being labeled. Here I'm showing you a picture. Back to on the top is that same transgene combination I showed you earlier, that paradigm-specific M cherry. And then I put underneath a fish that also had the KRT-GFP. And this is just to show you that this red transgene does not label every single cell, right? It's scattered, it's mosaic. The green does, but the red doesn't. And so how are we going to take advantage of this? So the important thing is that normally this transgene combination red doesn't do anything. But when we add a drug called metronidazole to the water, that kills some of the red cells. It doesn't kill all of them, but it induces enough extrusion events to where we're getting loss. Now, here's a movie that I took of, this is the tail of a zebrafish. I exposed it to MTZ and then just watch what happens. And you can see, you'll be able to see a lot of cells coming out. You see a bunch of cells here leaving the tissue. There's actually even already some tissue loss right at the edge, right, where a big swath of those cells has come out. Now, we can do this by live imaging, but maybe much easier, we can look at just static pictures, right? And so here on top, I have a wild type, where I have the M cherry, right, kind of mosaic pattern, the KRT-GFP, which labels the entire tissue, and then we have a merge showing both. And we can look at the bright field again. So we're looking, from here on out, we're going to be looking at just the tail of the fish. It's much more easier to visualize. And then on the bottom panel, we have when we add this dose of this drug, MTZ, metronidazole. And so what happens is that after 5 hours of exposure, you're getting a lot of these tissue loss, where normally where I have these arrows, you should see green cells, right, covering that entire tissue. But there's been so much damage from this drug that all these cells are lost. And now you have these gaps where there's no more periderm left, right? So that's really, we're trying to mimic this sort of chemotherapy phenotype you get. And so the important thing is that when we do this MTZ damage, it is not lethal to the fish, right? The fish don't care. Here I have multiple different doses. The dose I showed you in the pictures and in the movie is this 10 millimolar, this purple, and we still get about 90% of the fish make it. So it's really not lethal to fish. So we thought, okay, we have a great way of causing sort of widespread tissue damage, but importantly, we're not ripping off the whole skin, right? Because of the mosaic nature of that transgene, we're getting just a sort of salt and pepper-like loss of cells. Now, so we've effectively made this model where we can lose barrier function in the zebrafish embryo. And so now we can test, okay, are there opportunistic infections that we can test in this model that we have now? So for that, we joined forces with 2 professors here at MD Anderson at the Department of Infectious Diseases, Cotianes and Sebastian Worster. And so these 2 clinicians work essentially, they see patients that are in the clinic for cancer treatment, but then have some sort of opportunistic infection come up. And so they're able to grab patient-derived samples, right, of some of these opportunistic infections, these bugs that cause opportunistic infections. And so for the remainder of this talk, I'm going to focus on one of these, and that one is fungus, and the bug is called Rhizopus oryzus. And so this is a clinical isolate, right? We got it from patients, or they got it from patients, and we did experiments in fish. So we're going directly from the clinic into the lab, and hopefully back and forth. And so the assay we're going to do, so we have homeostasis, right? I told you earlier that not a lot's happening. We have some of these cells labeled in red, but nothing's really happening. What we do, we add this MTZ for 5 hours. And that is enough to induce this extrusion, right? So we're damaging cells. These red cells are getting pushed out, and then we're going to recover. And so after 5 hours, we wash out the MTZ, so get rid of all the drugs, and then just leave the fish, right? Now we take these fungal spores that we got from the clinic, and we've modified them to now they have GFP, so we can track them. So these are GFP-labeled fungal spores. We incubate them with a fish that was damaged, and we let them sit for 16 hours. At the end of those 16 hours, we wash that out, right? Wash out the spores, get them all nice and clean. And then we just wait, right? We watch for progression. What happens to the fish? Is it going to make it? Is it going to die? And essentially, we monitor for survival until day 7, right? So the end happens at day 4, and we're leaving all the way in day 7 to see how many of the fish made it or did not make it. So pretty straightforward. Now, what happens, I'm sure this slide might be a little busy, but the important thing is that this red bar here is what happens when you have epithelial damage and exposure to fungus. And so we're about 40% survival, right? So we've lost 60% of the fish. Now, importantly, if we add just Rhizopus oryzus, just the fungus, it's the blue line here, we don't see much of a drop. If we do just epithelial damage, again, we see about 90% of the fish make it. So there's no statistical difference between uninfected, just damage, or only infected. Only when you have the combination of damage and infection do you get this synergistic effect where you're killing the embryos. So we thought this is a really cool assay we have in our hands. Now, one really important thing and control that a lot of you are probably thinking about, well, is this dose dependent? And I will tell you in this slide that yes, it's dependent on the amount of Rhizopus that we add, right? So we'll have tissue damage, and it's dependent on how much fungus we add. So if we have tissue damage and then we add very little fungus, then the fish don't die as much, right? So it is dose-dependent matter. Now, one thing that I'm not going to show you today, but I can tell you that when we do the control for how much damage we do by doing different doses of drug, of MTZ, that also dose response. So it's a dose response of 2 things. 1, how much damage, and 2, how much bug is there. So they really do talk together. So for the rest of the talk, for my Rhizopus dose, I'm going to use this red bar, right, which is 5 times 10 to the 6 the spores. So what happens, right? If we do no damage and just add the fungus, what does the fish look like at day 5, day 6, right? So we do find some of these spores. Here's you're looking at the face of a fish. So importantly, this here is the mouth, or as you would think. And then these two circles up here, everybody thinks are the eyes, but they're not the eyes. They're actually the olfactory epithelium. So this is what the fish uses to breathe, to smell, right? So they're not it seems like it's looking at us. The eyes are actually way on the side of the head, and they're not visible in this magnification. But we're going to look at a lot of these olfactory epithelium, these pits. Now, one of the interesting things is that even though there's no damage and we add fungus, we do see spores, different areas in the fish. So the spores are able to sort of bind and hang on a little bit to these fish. And here's a picture of the tail. Again, in this picture, there's not many spores. It just happens that we didn't see them any in this fish. Now, what happens if we look at one of the fish that got damaged and the fungal infection? So this is much different. So number one, we do see these spores again, right? These round spores. But we see a transition of when before there were just a lot of spores, these round things. Now they've changed the way they grow. There's a bunch of hyphae, right? So we think that the fungus is actually sensing and is able to grab hold of something in the extracellular, something that's exposed because of the tissue damage, and that triggers a change in the way the fungus is going to grow. It knows it can stick around, so it begins growing differently. In the tail, again, we see a ton of these hyphae, right? So the growth of the fungus has gone uncontrolled now, and this fish is going to die soon. So we thought about, okay, we know fish. I'm sure you've heard some talks today about fish regenerating. If not, you're going to hear a lot soon, I'm sure. And so we really wanted to see, okay, can we exploit that for our good, right? Can we take something about fish being able to regenerate and find something out about it, right? And so we took our assay that we have normally, right? The 5-hour drug treatment to induce damage, wash it out, add the fungal spores for 16 hours, wash it out, and then monitor. We wanted to change it a bit and see if we could get at some of this regeneration aspects. And so how do we do that? So we do our normal tissue damage assay where we add the MTZ for 5 hours, and then we wash it out. And then what we do, we let the fish recover for 20 hours. No fungus, no nothing, just recover. After that recovery period, then we add the fungal spores, and then we monitor for survival. And so I wouldn't be telling you about this if it didn't work, but it worked beautifully, right? So if we look at this graph, if we do our normal assay where we do damage followed by fungal exposure, it's this red line here, right? So we end up again with about 60% lethality or 40% survival. But if we give the fish 20 hours to recover, we see a pretty good increase in recovery now. So I'm sorry for the green and red, but we're up to about 75% survival. So we're actually pushing back, right? So this tells us that if we give the fish just a little bit of time to recover that tissue damage, that lack of barrier function, it really means a lot in survivability, right? And so we're really interested in figuring out how does the fish do this. And so we decided to focus for the rest of the talk and the paper essentially on this time frame, right, of the metronidazole and the recovery. So let's forget about the fungus for a little bit. Let's figure out how does the fish recover, and maybe we can learn something about epithelial biology that we can apply to the clinic or to something else, right, which is really important. That's really what we're here for. And so these time points can be two things, we think. One, you can prevent damage, right? So if you prevent the damage or the amount of cells that are released, then you're better off, right? You're not going to have as much loss of barrier function, and you'll have more protection. The second thing is maybe you're accelerating wound healing, right? And so we wanted to sort of figure out which of these is a fish doing and which of these is responsible for us seeing an increase in survivability when we give it just a little bit of time to recover. And so we wanted to see what genes are involved in this, right? And so we did a classic experiment that everybody does when you don't know sort of where to go next, right? We did RNA-seq, and so we did homeostasis, so no damage, versus just the damage. So what genes go up just in response to damage? And we found one of the highest regulated genes that go up on damage is something called epigen. I had never heard of an epigen before, but when I looked it up, it turns out to be an EGF-like molecule. It binds through EGF receptor. So we thought, okay, EGF, right? Could matter. Could be something important. So just to give you an idea of a refresher for those of you guys that took developed biology a while ago. So EGF is a ligand, right? In this case, green, that binds to the receptor, EGF receptor. Sorry, my microphone. There we go. To EGF receptor. Oh, sorry. I don't know what I did. Oh, there we go. The EGF receptor, right? And what happens is that when it binds, you lead to a signaling cascade, right? To a whole bunch of different things, and growth and all kinds of stuff that we're not going to talk about today. And so all you need to know about today is that epigen is very similar to EGF, and that also binds EGF receptor. And so the first thing we did, we wanted to see, okay, is this thing even real? We found it by RNA-seq. Let's do some in situs, right? Fish are easy to do in situs in. And so we did no damage versus damage, and we just did an in situ for epigen. And it turns out it's expressed under no damage. You can see in this picture here, there's some cells that do have each epigen transcript, but then after damage, it really goes up, right? So that says that our RNA-seq probably was right. Now, the next thing we did is because fish are so easy to put drugs into the water, we said, well, we had some EGF in the freezer, so let's just dump it on the fish, see what happens. And we also ordered epigen just to see. So we're trying to do two things at once, right? So we added both. And now, again, why zebrafish? Well, it's really cool. You can put them in these little dishes, and you can multiple fish in one. But also, you can have these 4-well plates where in each different well, you can add a different molecule, something different in each one, right? So you can add EGF in one, nothing in one of them, epigen in another, and then just watch and film. And so here's some movies of what happens. So homeostasis, obviously, there's not really much happening at this time. Now, the MTZ treated, you'll see a lot of those contractions and then a bunch of tissue loss, right? The cells just go crazy and get out. But when we add EGF or epigen, we see those same contractions, right? But the cells never extrude. They never get pushed out. So they're probably getting damaged, but just something about the tissue is not letting them leave. So we thought this is super interesting. We didn't think we could uncouple the pulses from the extrusion, but it seems like we did in this case. And so, sorry. Stuck on the movie. There we go. Yeah, so when we quantify this, when we add EGF or epigen during MTZ treatment, this suppresses the amount of cell loss. So we count how many cells are extruded. If we have just MTZ treatment, we go from, in 3 hours all the way to about 12 or 15 at 5 hours after damage. But if we add epigen or EGF, we can really bring that down to almost 0. And again, the wild type, we never see any extrusions because nothing's happening at that time. And so now comes we saw this thing, now comes the real experiment, right? So if we add epigen or EGF to the fish when they're getting MTZ'd and then expose them to fungal spores, what happens, right? This is what we're doing. I mean, it's cool with cell biology. I love cell biology. But at the end of the day, does this help with this opportunistic infection? And again, I wouldn't be telling you this if it didn't work, but it did work, thankfully. And so we see the blue lines in both of these charts are controls where they're getting damaged and they're getting exposed to fungus. And again, we're seeing about 60% of them die off at the end. But if we add either EGF or epigen, we see a drastic increase in survivability. Now, one important thing is that if you look at this EGF, right? EGF, you get about 80% survival. But the important thing is we didn't identify EGF in the RNA-seq, right? We identified epigen. And if you do epigen, you get about 85, 90% survival. So we think something about the binding affinity of epigen versus EGF itself is something driving this whole thing. So we're super interested in how that happens. Now, we think that either normally epigen and probably also EGF work somehow to prevent this extensive tissue damage loss, right? So there's somewhere sitting there preventing this extrusion events or many of these events from happening. Now, how they do that, we're not entirely sure. We have some ideas. We're still not worked out very well for us. And when you do that, you prevent the fungal spores from coming in, and then you increase your odds that you're going to survive, and that's why we see an increase in fish surviving when they get either epigen or EGF. Now, one super important thing that when we started looking around after this, we found there was actually a clinical trial going on, right? And so I'll read it to you. I don't like reading slides, but I'll read this one. The purpose of this study is to determine efficacy and toxicity of recombinant human epidermal growth factor, which is EGF, as preventive drug of oral mucositis during intensive chemotherapy. And so the important word here is preventive, right? Our studies showed we did the multiple things. We did EGF during infection, EGF during the MTZ, EGF before the MTZ. We did all these different things. And it turns out that when you do EGF either during the infection, so after they've already gotten tissue damage, or during the tissue damage, they both work better than if you do the EGF treatment as a prophylaxis treatment. If you try to give it to them before they get damaged, it doesn't work as well. So just in our study, I think we uncovered something that they may have missed, right? They tried to do preventative. Maybe it was better if they do it during, right at the time they're getting the damage or the treatment, or maybe after they got chemotherapy. So just by using zebrafish, we've already found something new that can immediately be applied in the clinic. So what are we doing now, right? So we're really interested in genes that maybe enhance epithelial restoration, right? So once you get damaged, how can you get that damage closed fast enough? How do you get around these things? And to that, what cells are responsible for doing this? We really don't have any idea. We know we have a basal layer, and we know we have a periderm. We assume the basal layer is the one that divides and repopulates that periderm, but we don't know, right? So we're doing some single cell RNA-seq. We're trying to figure out what cells are there. Are there cells that may be more important for this response than others? What does a real stem cell look like? Are all stem cells created equally? We really don't know that, right? We're also super interested in antifungal drug discovery. So now we have this cool assay, right? We can damage the fish, add the fungus, and then see what happens. Well, what if we add a drug with it too? For our paper, we added a couple of drugs that they use in clinic, right? We use Mucosal. That's one that's used every day in clinic, and it works in our assay. I mean, it's not surprising, right? It works in the clinic, it works in the fish. But can we look at other things that aren't being used yet and maybe find something even better? Right, so we're really interested in drug discovery. And again, this is what I just told you, right? This is one of the things that we did for the paper to show that our assay actually works really well. And so we're interested in doing this. And I told you, zebrafish, you can do high throughput, right? You can do hundreds of molecules at one time. So we're really interested in that. One of the cool things we're interested in is we know that after extrusion happens, we know that these cells that are remaining, they secrete chemokines, right? They secrete these molecules that signal to other cells around, like, hey, something's happening here. And we know that when you do the damage, so here I'm labeling neutrophils. I'm using a transgenic line where you label neutrophils. And when you have MTZ, right, when you have tissue damage, you see these neutrophils just go everywhere. So they're surveilling the tissue, and they're finding out what happened, where the tissue lost, what can I do? Now, importantly, if you suppress the tissue loss with epigen, you don't see as much. So you're actually lowering down the amount of the immune system working, right? So we think this could be important for neutropenia, for all kinds of other things. So we think that's a really important and cool angle to study. And then one of the last things we want to study is retention of damaged tissue, right? So I'm telling you, you can see in this movie at the bottom, when we add MTZ, even with epigen on there, we're still getting some tissue loss. So what's happening there, right? Do we want that tissue to sit around? So let's say we have this tissue and we have epigen there to keep it around. If we add more, are we keeping sort of these undead zombie cells? It's October, so it's perfect, right? Halloween, right? So we think that by suppressing this, even though suppressing this this extrusion response may be really good short term, we don't know what that means long term. We don't know how long those cells are staying there. Maybe they're there only for a couple of days and then kicked out. We really haven't looked at that. So we're really interested in what happens where these undead cells are sticking around in the tissue. It can be good, right? Things like that. That's how you get cancer. You have these damaged cells sitting around that don't leave the tissue, and then maybe they divide, and then you leave with something like cancer, right? So we're really interested. We don't want to cause more cancer by trying to prevent an opportunistic infection that's happening because of the treatment. So just really quick, I want to acknowledge the lab, right? Everybody that did this. So I've moved on from the lab. People moved on from the lab. There's an old picture. So down here at the bottom, Yumna, I showed you that cool extrusion movie when they're pulsating. That's her movie, right? I took it from her, or borrowed it from her. Here at the bottom, I have a little GIF of these fungal spores that are transitioning from being just spore into hyphal growth. And then people that helped with this story specifically. So Kristen did a bunch of the quants for us. She was really helpful. Philip Kahn is the one that did all the fish work, essentially. Well, all of the setting up of the fish, and then I would show up the next day and have fish ready to go. So he did a lot of work for me for the project. Kim Ann also was really good. Was also super helpful in letting us use one of the scopes in her lab to use for imaging the fungus, right? We didn't want to have the fungus in our main facility where we have fish. So we went to a place where they do a bunch of just fungal stuff and sort of host-pathogen interaction stuff. And that they're more open to having not good fungus around. And of course, in the Cotianes lab, Dr. Cotianes, the PI, and of course Sebastian, my partner in crime in this. This whole work was done thanks to a CPRIT grant during my time at MD Anderson. I'm super grateful to the lab, especially to my mentor, George Eisenhofer. I learned so much from him. Now that I'm on my own, it's really daunting, right? You're training with somebody, and then you go off and you start your own thing. It's crazy. And so if anybody want to contact me, this is my Twitter handle, in the OR. I'm happy to chat about more stuff. If you have questions, I'm happy to do anything you guys want. I do a lot of science communication and things like that. And I love imaging, so I'm happy for anything anybody wants to talk about. I post a lot of jobs, so that's all I do. And on Twitter is I repost jobs. And then if you hear somebody snoring, I'm sorry, it's not me. It's my dog who's sitting behind me. But with that, I'll stop. I'll take any questions. And thank you for the invitation. I'm super grateful. I'm glad to be here.
Thank you, Dr. Oscar. It was a great talk today. We have some questions in the chat. I will ask you first from Nicolas Trigueiro. He asks, it is possible that aesthetic fission is one of the mechanisms involved during the wound healing to cover the hurt area quickly to avoid the loss of barrier function.
Yeah. So we've done a ton of live imaging, right? And we don't see a lot of that happening, right? So we think what we have caught more is cells from the bottom layer sort of migrating up to the top layer. So we have not seen, I'm trying to think, I don't think I've ever seen. So one other thing we do see is cells getting big, but never them fusing, right? So that's a great question, but I haven't seen it yet. It doesn't mean it doesn't happen. It could totally happen. We just haven't caught it.
Okay. Thank you. The next is from Enrique Salas. He says that's an excellent talk and asks, damaged cells lose or down-regulated cell adhesion proteins during early development. E-cadherin is regulated downstream of EGF.
Yeah, that's a great question, and that's exactly what we think, right? So we think that when you have this EGF during this tissue damage, we think that what happens is you're making the tissue stiffer, right? It's making it stiffer, and that allows it for it to stick around. Some of Yumna's work in her paper are both on that same edge. This is things happen, and we think it's more of tissue stiffness. So Yumna made the tissue stiffer. She actually measured it by I'm trying to think of the name of it's force microscopy, where you can pull. It's like a record with a needle, and you push down different areas. And when she did it on fish that were treated versus non-treated, treated with damage, right? She sees different viscosities of the tissue, and we think that that's a main thing that we haven't done that experiment. Didn't treat it. We really want to do that. We also want to do. I showed you that cool movie she had, right, of the movement. We want to measure the amount of tension by doing laser ablation and see the amount of recoil. Is it really the stronger, the junction is stronger or not? That's exactly what we think is happening. Great point.
Okay. And the last one is from Maraisa Mello. She said, thank you, Dr. Oscar, beautiful images and videos. Did you check the cytoskeleton on the cells that extrude the neighbor cell? How the cytoskeleton looks like on those cells when you treat it with EGF?
Yeah, that's a good point. I haven't done that. We did it by live imaging. Problem is with live imaging, well, two problems. The problem with live imaging, you can't really get at a nice, right? You can't really get in there. It's like 60x. But I think the bigger problem is that when you do the treatment, you're not getting a lot of extrusions. So it's hard to go in there and find the extrusions. We can do it. We just haven't gone around. We rely, I think, too much on our life actin GFP, which is the GFP I showed you. But we can do tubulin. We can do all kinds of stuff to see what happens. Yeah. I suspect that the actual mechanism by which extrusion happens probably is the same, but it's just that the tissue stiffness is different. That's what we get as much. But great question. Yeah.
Okay, I think we don't have any more questions in the chat. Dr. Oscar, tell us his Twitter. It's on the chat in the put it in the beginning of the talk. If you have more questions, you can contact him, I think.
Yes, anytime.
We'd like to thank you, Dr. Oscar. It was a great talk, and thank you for your contribution today. And now we will give another break, and we will put our sponsors video. So we will be back soon. Thank you.
Thanks, guys. Muito obrigado.
Obrigado.
My name is Mark Francis. I'm the founder of Aquaneering. For the last 20 years, Aquaneering has built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community, and we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us, because for Alesco, research is for life.
Welcome everyone to FST's presentation. We appreciate you taking the time today and look forward to helping you find the right tools for the right application in your endless discoveries and research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.
When you walk into a pharmacy, over 90% of the products on the shelf have been tested on animals, such as rats, rabbits, and primates. These animal models have the complexity one needs, but they are costly, time-consuming, and ethically challenging. Also, 9 out of 10 products tested in animals fail when administered to humans. Even with biotechnological advances, the use of these animals in product research and development is still massive. Is there no other alternative to the use of these animals? And to what extent is maintaining this model worth it? Today, regulatory bodies pressure companies to change this scenario. In this perspective, large companies such as Natura, Boticário, and others are adopting an alternative methodology to replace the use of animals in tests. Thus, to offer innovative solutions, ZSafe was born. We are committed to using fish chambers for the toxicological evaluation of new products. It is an innovative, alternative, and predictive model. The fantastic thing is that, despite not being considered an animal model, this embryo shows cellular activity, guaranteeing the same reliability in animals and greater than tests in cells. Our tests reduce research time and costs in new product development by more than 50%. We have the perfect formula to provide greater profits for companies and reduce any possible ethical issues regarding animal use and welfare. So it's a win-win relationship. Our differential in the market is the competitive price, shorter service time, and delivery of reliable results. We have a multiple competent team that focuses on innovative solutions for the market and works to generate biomedical research safely and efficiently for the good of our customers, society, and animals. We are disease safe, and we are swimming towards a safer future.
My name is Mark Francis. I'm the founder of Aquaneering. For the last 20 years, Aquaneering has built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community, and we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us, because for Alesco, research is for life.
Welcome everyone to FST's presentation. We appreciate you taking the time today and look forward to helping you find the right tools for the right application and your endless discoveries and research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.
When you walk into a pharmacy, over 90% of the products on the shelf have been tested on animals, such as rats, rabbits, and primates. These animal models have the complexity one needs, but they are costly, time-consuming, and ethically challenging. Also, 9 out of 10 products tested in animals fail when administered to humans. Even with biotechnological advances, the use of these animals in product research and development is still massive. Is there no other alternative to the use of these animals? And to what extent is maintaining this model worth it? Today, regulatory bodies pressure companies to change this scenario. In this perspective, large companies such as Natura, Boticário, and others are adopting an alternative methodology to replace the use of animals in tests. Thus, to offer innovative solutions, ZSafe was born. We are committed to using fish chambers for the toxicological evaluation of new products. It is an innovative, alternative, and predictive model. The fantastic thing is that, despite not being considered an animal model, this embryo shows cellular activity, guaranteeing the same reliability in animals and greater than tests in cells. Our tests reduce research time and costs in new product development by more than 50%. We have the perfect formula to provide greater profits for companies and reduce any possible ethical issues regarding animal use and welfare. So it's a win-win relationship. Our differential in the market is the competitive price, shorter service time, and delivery of reliable results. We have a multiple competent team that focuses on innovative solutions for the market and works to generate biomedical research safely and efficiently for the good of our customers, society, and animals. We are disease safe, and we are swimming towards a safer future.
My name is Mark Francis. I'm the founder of Aquaneering. For the last 20 years, Aquaneering has built a team of dedicated professionals to support your research. Our mission is simple: provide the highest performance zebrafish housing at a reasonable cost. Our team is here to allow you to focus on your research.
We at Alesco work daily to create solutions and equipment for the scientific community, contributing to the advancement of Latin America biomedical research. Our mission is greater than just providing equipment. It is to provide security with a quality service and mainly with enormous respect and attention. For this reason, we invest in high technology and seek to maintain long-lasting relationships. Our commitment is to be a reliable partner that understands the needs, the reality, and the conditions of each customer to always offer the best solution. We understand the benefits of scientific research for humanity, and this encourages us. If today we have a better quality of life, greater longevity, if we beat cancer or use a medicine for a headache, it is because the advance of biomedical research allows us to. We trust in the work of researchers, in science, and the scientific community. And we pride ourselves on doing our part. Just like you, we are passionate. Science is what moves us. Because for Alesco, research is for life.
Welcome everyone to
Hello, everybody. We're back for our last presentation of the day. So we're very sorry for the delay because we're having technical problems, but here we are now and we're going to finish very, very nicely. I'm going to invite Bianca Ventura, our dear friend, to present our last speaker of this day. So Bianca, here's the words are for you now.
Thank you. Hello, everybody. My name is Bianca. I am from University of São Paulo. And first, I would like to thank everyone for attending this virtual webinar. I would like to thank Mike for accepting the invitation and speaking on this very relevant topic for those who work with zebrafish in research. Professor Michael Kent is a veterinarian at Oregon State University. His line of research is related to fish disease, parasitology, and pathogens that affect zebrafish and salmon. And today he will talk about effects of underlying pathogens and the disease on research endpoints in laboratory zebrafish. Now I will give the floor to Dr. Michael Kent, and you can send the question in the chat that after the class we will be able to answer. Thank you, Mike.
You're welcome. Thanks for the kind introduction. I'm assuming you guys can see my screen now and everything. I'll put it on the actual presentation mode. Can everybody hear me and see the screen? Can you hear me?
Yeah. Yes. Okay. Yes, Mike. Now it's okay.
All right. Everything is okay. Okay. So thank you all for sticking around today to hear. I know it's much later in many parts of the world that are hearing this, and I'm going to be talking today about review of diseases and impacts in zebrafish facilities. And this is really based on research that we've been doing for the last 20 plus years here at Oregon State University. That's where I'm at. And then my colleague, I have other colleagues as well too, Dr. Katie Murray, who's a veterinarian, PhD down at University of Oregon. So that's down the road about an hour from us. And that's where the Zebrafish International Resource Center is. And I'm a co-principal investigator with them. I've been working with them since they formed in 2000. And so a lot of the work that I'm going to be talking about today based on the demographics or prevalence, distribution of infectious diseases and a couple of others is based on the diagnostic service that we've been running. And then I'll be talking about some of the research that we've been doing and others to look at the impacts of research. So I'm funded by the Office of Research Infrastructure Programs, and so is the ZIRC. We call it the ZIRC, Zebrafish International Resource Center. We call it ZIRC. I know you guys down in Brazil will call it ZIRCI. But anyway, it's funded by the National Institutes of Health, and they fund both the center as well as my research up here at Oregon State University. So this is actually a talk that Katie Murray gave about a week ago. Both she and I, she presented this. She gave a 15-minute version of this to NIH, and I'm going to be giving you the longer version, about 40 minutes. So it's rather surprising that diseases in laboratory zebrafish, that we have that many pathogens, that many infectious diseases. Because we know the way that they're reared is in very defined water systems. We have often reverse osmosis water that's treated, so we're not exposing them to a lot of pathogens. But regardless, we still have a lot of pathogens in them. And I'll be talking as I go through my talk why some of my thoughts on why we still see a lot of diseases in them. So the list of bacterial diseases: Edwardsella, cholerae, Flavobacterium, Mycobacteria, various species. Mycobacteria are the important. Parasites, Microsporidia, Pseudoloma is actually in about 50% of the labs, as well as another Microsporidium that's not as common. We even have metazoan parasites, worms, myxozoans, etc. That's rather surprising for inside of a university setting, but we do, and a few other parasites. We also have viral diseases, and this is more recent. As far as viral diseases, ISKNV is very important. This nervous necrosis virus. These are cause significant disease, but are very rare. And then, in contrast, just recently we recognized that there's a coronavirus that's in about 50% or even more of the facilities, but doesn't seem to be that pathogenic. And then we move on to the list of non-infectious diseases, such as water quality problems, which are actually can be devastating. Like if you get chlorine in your facility, or other related problems, malformations that we call idiopathic. That is, we don't really know what the cause is. GBD, I'm going to be talking about this. This is gas bubble disease. EAIF, egg-associated inflammation and fibroplasia, and hepatic megalocytosis are just part of the list. And then we have a lot of spontaneous neoplasms in zebrafish. Seminomas, intestinal tumors, chondromas, peripheral nerve sheath tumors, that's PNST, and ultimobranchial tumors are some of the most common ones. I might mention that I'm going to be talking later in the talk, we say neoplasia, we can put the intestinal tumors or intestinal carcinomas, we could put those in the category of infectious diseases. We don't know for sure what the etiology is of these intestinal carcinomas, but we know that they are transmissible. So again, here's the Zebrafish International Resource Center down in Eugene. It's a standalone building that was largely funded by National Institutes of Health. It's been going on there for about over 20 years, since 2000. They provide, the most important function is providing a number of zebrafish lines and wild type fish, etc. Katie Murray is the attending veterinarian, and I work very closely with Katie. And we've been running a diagnostic service since 2000 with that, which we provide a diagnostic service to the zebrafish community. So we're looking at about up to, we always have to update these. It's three years out of date, so then we can kind of bump the numbers up a little bit. We probably looked at 17,000 fish from about 330 or so laboratories. And here's the breakdown on some of the diseases. I'll be bringing this graph up periodically because it puts things in context. Pseudoloma, close to 50% of the facilities have this problem. That's followed by mycobacteriosis at about 40%. And then we have a variety of other pathogens, a myxozoan. I'm not going to be talking about this today. It's actually pretty common, but it's not that pathogenic. I'm going to be spending a fair amount of time talking about Pseudocapillaria tomentosa. This is a nematode, an intestinal nematode, a parasite that's quite pathogenic. So these are the main ones I'm going to be talking about, and a couple of others as we go through as far as the non-infectious diseases. So these are the infectious diseases or pathogens. So let's first talk about mycobacteriosis. So it's a disease that's caused by multiple species. And I'm going to get into this a little bit to talk about concentrating on the endpoints. So when we think of clinical disease, we see emaciation in fish. The hallmark, we often think of mycobacteriosis as causing ulcers. So we have swollen bellies, emaciation, as we see here, skin ulcers, general malaise, exophthalma, their eyes may be popping out. And when you open the fish up, the hallmark of mycobacteriosis is the presence of granulomas. And here's one. The zebrafish are small, so you have to really look at these under a dissecting microscope before you could really appreciate these. And this is a severely affected fish with lots of granulomas. A lot of times they're better appreciated by just looking at histology. So a focus of my talk today is on what are the impacts. And obviously, the first impacts are, these are chronic inflammatory lesions. They're caused by a number of species. I'm going to get into this in the next slide. Asymptomatic to high morbidity and mortality. So those are just straightforward clinical disease. Then we have other things. I'm going to talk about mycobacterium causing the granulomas being autofluorescent. And that's confounding non-protocol induced variability if you're doing studies where you're looking at fluorescently labeled objects in your fish. Human infections. I'm going to be talking about this a little bit. There's been 2 laboratories where a number of people have developed mycobacteriosis from working with the zebrafish. The way they present is often granulomas, but they could be discrete granulomas to just sheets of diffuse chronic inflammation. We can have acute infections, chronic infections. There have been more recent, there have been papers that are showing, and no surprise, with all this chronic inflammation, we're going to see effects on the transcriptome signatures. And we have some work that Justin Sanders has done, not been published yet, on inflammatory cytokine alterations in presentation of cytokines. So taking a little step back, we always say mycobacteriosis, it really is you could really divide them into 2 very different diseases with 3 major species. The first is Mycobacterium coloni. This is by far the most common infection. Many fish are asymptomatic, but they still have lesions if you open them up. There'll be individual clinical cases. So if you have a large population of fish, a few of them will die with coloni. You don't see big outbreaks. But but we're talking about laboratory models, so we don't just dismiss this, because this is the one we're going to be concerned with underlying chronic inflammation, granulomas affecting other research endpoints. Then we have Marinum and Haemophilus. These are very acute, highly pathogenic infections. You're not going to be worried about them affecting research endpoints because they're going to be killing all your fish, and you're going to have to get rid of the fish and start, remove the fish and start over. So we have Marinum, very pathogenic to zebrafish, and this is one that's a zoonotic concern in particular. At 2 research facilities have had a number of people where they had Marinum outbreaks where staff actually developed Mycobacterium marinum infections. Rare, only 2 major outbreaks. There have been a few other incidental ones. And then Haemophilus, we see this more often. It is highly pathogenic. It's more chronic, but it still is lethal with massive numbers of bacteria in the fish. So a focus of my talk are subclinical impacts. So here are granulomas, and Dr. Whips, Chris Whips, my former student, he's at SUNY University doing independent research. He published a paper in 2015 with colleagues showing that these granulomas actually autofluoresce. And so if you have a fish that you're putting in a tumor, let's say, that's GFP labeled, this is going to confound your studies if you have these background autofluorescing lesions. And again, as I said, influence on cytokine expression. This is some work that's not been published. Here what I'm doing is tracking the amount of inflammatory tissue in a section of the zebrafish, and Dr. Sanders is taking the other half of the fish and is looking at changes in endpoints such as IL-10, etc. Not a surprise that it's going to be making changes in these fish. So if your research endpoints includes these, your research includes these kind of endpoints, you do not want to use fish that have underlying mycobacteriosis, even if they appear clinically normal. Now this is quite important. Nobody died from this infection, which is important, but it caused some problems to the laboratory. So think about mycobacteriosis, again, direct causes of mortality, contributing to mortality. If you don't get contributing cause of mortality, morbidity, non-protocol induced variation, that's what I talked about. What about egg production? unknown impacts on experiments, many experiments. I showed you some examples. I'm sure they go beyond that. This is important to consider in a zebrafish facility. Mycobacterium marinum is a zoonotic concern. We know this very well, people working with tropical fish, handling fish, getting these hand infections. Fortunately, it does not go systemic. It can be disseminated in the skin, but does not go to systemic. And the people that get these infections have to go on like 6-month programs with antibiotic treatment. So this can be a very big problem. And you can think about the liability at your university. If you have fish infected with Mycobacterium marinum and you send them to another facility, and at that other facility you have immune compromised people that get nasty infections by this, your university, at least in the United States, the university would likely be responsible. And I'm sure that the administrators of the university would not be happy with you if you cause disease problems in your staff as well as other staff. So do not, the concern of marinum goes beyond just killing fish and non-protocol induced variation in experiments. Now this is one that Bianca knows very well. We've worked on a collaborative project together with this, and she can tell you a lot about the concerns and difficulties with doing research when you have these infections in fish. Infections by Pseudoloma neurophilia. It's a microsporidium. You know, so microsporidia, we've long thought of them as protists, but they're actually sister to the fungi. That's a taxonomic story. What's important is that they're obligate intracellular pathogens. They're highly infectious. So you don't see them growing in the environment on biofilms and things like that, like mycobacteria. So why are they so common in zebrafish facilities? Horizontal transmission, I said they're how the spores, I talked a little bit about the life cycle as they go through, are highly infectious. But this, we believe, is a key problem, is that vertical transmission outside the egg and in the egg has spread this parasite to all around the world. 50% of the facilities have them. The disease infections that they were first called skinny disease. So fish become thin, emaciated. Some of them will show skeletal deformities. And then if we look at different organs, particularly the central nervous system, meninges, here's a nerve root, we see aggregates of these spores. I'm going to show you a couple of other pictures here in a minute. So what is associated with weight loss, skeletal deformities, mortality, and immune compromised fish? I'm not going to, I have limited time today to get into this. Most fish are relatively asymptomatic, but nowadays we're using gamma radiated fish to transplant human tumors and other tumors into them. And Dr. Sean Spagnoli showed that mortality, if you have fish with underlying microsporidiosis and you gamma irradiate them, you severely immune compromise the fish, then the parasite can become more pathogenic. And then this is some interesting paper by this group, Midtun in Norway, just a couple of years ago, showed downregulation of immune response genes suggesting immune evasion strategies of the parasite. So if you're looking at transcriptome, particularly immunologically related transcriptome endpoints, having underlying infections by this could be causing, could compromise your results. Again, I showed you the slide earlier. Keep in mind how common this parasite is. Skinny disease, as I said. And here's a picture, another picture showing a typical presentation with aggregates of spores along the central nervous system, more minor meningitis, the equivalent to meningitis in this, but sometimes this can be quite profound, as I showed in an early one. So a variety of lesion presentations. And if you squash these out, look very careful under the microscope, you would see the microsporidium spores. They're very tough. They have a posterior vacuole. They're about 5 microns long. This is a 10 micron bar here. Very distinctive morphology. So all these little dots here, this is a much lower magnification. We went to a higher magnification, removed this and put it under phase contrast, the Marsky's phase contrast. You could see these individual spores. But most of the time our diagnosis is done by PCR, histology, special stains. Here's a special stain. This is actually acid fast, so we use for mycobacteria. We'll stain these, but a Luna stain will show similarly. I'm showing the special staining of the spores, but also want to point out that another presentation, the name is pseudoloma neurophilia, indicating the infection of the central nervous system, but it will also infect the muscle, causing significant myodegeneration and myositis. Let's talk about the transmission a little bit. Of how I just briefly mentioned that it was vertically transmitted. Dr. Justin Sanders did a big part of his thesis with me a number of years ago, was looking at transmission. And here's another special stain you could use with microsporidia, a gram stain. And here is a zebrafish. Here are zebrafish developing eggs, and here are these blue staining spores, gram positive. Here's an individual spore. So not only do they occur in large numbers in the ovaries, in the stroma of the ovaries, where we can see up to 100,000 spores in the ovaries. So even if the fish are spawning, they'll just be spewing the eggs out. They actually occur within the eggs. And here's a developing egg, developing embryo. We follow this white blob here. This is not normal. Here the embryo is developing more. The same embryo, Dr. Sanders followed this. We see these two blobs. Here it is, almost fully developed, not hatched yet. These two white areas. And then at that point, he killed the fish, squashed it out, and you can see those white areas are filled with microsporidium spores. So you can see no matter what surface disinfectants you're going to use, you can't eliminate them. So here's a review that was written. This is following on Dr. Sanders' work 10 years ago. Caballero Huertas did this paper a couple of years ago, summarizing the different modes of transmission. So you have actively horizontal transmission feeding on spores that are released in the water during spawning, carcasses, the traditional way pathogens are transmitted. But he's also showing this intra-ovum and extra-ovum transmission, spores that are released at spawning and actually spores within the embryo. Okay, so that's kind of giving you the background on pseudoloma. I'll talk a little bit about impacts. So you think about a central nervous system infection, what would be one of the first things you think about? How is it going to affect the fish's behavior? I've done some work with toxoplasma. Many of you that work in veterinary medicine are very familiar, or human medicine are very familiar with Toxoplasma gondii. In fact, I know that Brazil, it's quite common down in Brazil. So think of this infection sort of like toxoplasmosis in a zebrafish. We know very well that toxoplasma affects behavior of infected mice and rats, and probably even humans, central nervous system infection. So let's think about the zebrafish. As I said, if you're not immune compromising the fish, most infected fish do not exhibit clinical disease. They may look like this. So then people would go on to use them for their behavioral research. I'm not going to go through all the different aspects, particularly the follow-up paper by Mid-Tun et al. This group is behaviorists in Norway, and they've done some excellent work that followed up on Sean Spagnoli and our work here at Oregon State University. So Sean wrote a paper, and this is a spelling error. It should be E. It's not San Spagnoli, it's Sean Spagnoli. Mapping the areas of the brain where you see most of the infection. And these areas corresponded to, I'm not a neuropathologist, the greasum centrale, ventral white matter, reticular system, etc. And these are all involved in response to stress, startle stimuli, etc. So he went on and did some experiments to see how these infections altered startle response. Habituation habituation is inhibited. Shoaling behavior is inhibited. We also found that they avoid capture more than uninfected fish. So you're going to be, if you're netting fish out of the tank, you'd be biasing the sample. And then others, the Mid-Tun group went on and found reduced locomotor activity. And these changes were sex specific, etc. So it's quite a variety of behavioral endpoints that this zebrafish, that pseudoloma affects. And so about back in 2016, Nature News reported this. Petra Ziglagi, we didn't write this. This is her article. Parasitic infection may have spoiled zebrafish experiment. Common contaminant may change shoaling behavior or proxy to stress, anxiety, and behavior. So this made the news, at least the news in the scientific community. And that was based on this paper that Sean wrote, where he's showing average intra-fish distance was significantly increased with the infection. You look at this, I think I got a little time to talk about this. You go, okay, it's significant, but not that much. This is actually a greater or more reduced intra-fish distance than if you put alarm substance into the water. Alarm substance is what's released by the fish's skin. There's actually alarm cells, and that tell the fish that something's bad. You know, one of their tank mates has been eaten, so they crowd together in a shoal, and the response with being infected by pseudoloma was actually greater than if you expose fish to alarm substances. And then here's just another one of his first papers, and here's that paper that Helen Midtun and co-workers in Norway wrote. This was in Scientific Reports showing these different changes. So the behavioral stuff, you can see, it's so obvious to me that that would be a big thing, but I don't think the behaviorists really embrace this. They still go on and do their experiments using infected fish. Other endpoints beyond just morbidity, that the infections can cause, just like Dr. Whips's thing with the autofluorescent granulomas with mycobacteria, we found fluorescent nodules or granulomas with pseudoloma. This was with Kimball Fraser and Kylie West. I was a co-author on it as well too. They're studying cancer, and they realize this is a background problem. Again, they're trying to track fluorescent cancer cells, and their fish are looking like this. We did histopathology on these nodules or granulomas, and we find that they're actually caused by pseudoloma. So here's actually the pseudoloma within this granulomatous lesion in the muscle. Again, this idea of the muscle infection, we still haven't quite figured out why we see some fish, some populations, we see a lot of muscle infections. Other times we see minimal muscle infections. So, and why this actually happens, we don't really know. But this is another confounding variable. But can you imagine if you're trying to track tumors in fish and you have all this background in them, you're going to have some problems. Some of the basic husbandry stuff, moderate reduction in weight. This is with a group studies we did with TUs. And this is a study, we have a couple of other studies that would complement this, that sometimes the infection, these are infected fish, these are uninfected fish, will impact reproduction. Remember, it infects the gonads, etc., and it affects behavior. So it can have an influence on reproduction. Very variable. Sometimes it does, sometimes it doesn't. This is a pretty extreme example that I'm showing you here. Okay. I assume we're going to have questions at the end, so I'll just roll on with the next couple of diseases. I think that I was allocated about 40 minutes. I will get done, and I'll have time to answer questions. So here's another. It's just happenstance. It's just a curiosity that the second most important pathogen in zebrafish also starts with the genus name of pseudo. In this case, it's not pseudoloma, it's pseudocapillaria. The species name is pseudocapillaria. It's tomentosa, originally described in carp and goldfish, etc., in Europe. And so we see it in a lot of cyprinid fish, minnows and things like that. So it's a capillary nematode. If you think about those of you in veterinary and human medicine, the closest relative would be whipworms. And the infection, as they go through this, it is a lot like whipworm infections in humans. It's directly transmitted. There's no evidence of vertical transmission. Typical nematode eating eggs in the environment. What does it cause? Emaciation, clinical disease, as we see here. It is more pathogenic than pseudoloma. Moderate to high morbidity and mortality. There's evidence out there that AB strains seem to be more susceptible. It's like whipworms. It's a tissue invasive worm that induces a severe inflammatory response. So they're not just floating around in the lumen of the gut. They are actually invading tissue. So here's an intestine. Here's that egg that they eat. A larvated egg takes about 5 days before they larvate, 5 or 6 days. And we see all these worms in the intestine of a zebrafish. And I'll show you some histology to show that they're actually not actually invading the epithelium, and deeper sometimes. So tissue invasive. This is our group here, led by postdoc Galki. Microbiome distribution. No surprise with all the inflammatory changes that there's microbiome diversity correlates with parasite burden and disease. And a lot of work that I've been kind of leading with colleagues is promotion using the zebra that the worms are promoters for intestinal carcinomas. So here's what the picture I showed you. Here we show all these nematodes, and then up the upper right is an egg that would be collected from the environment, because it has to be in the environment for about 5 days. Here are some fresh eggs that you would find in, being freshly released. Here's a female. These would be freshly released, so they're not larvated. This is what you would find in the intestinal lumen of the fish. How common is this? Fortunately, only not nearly as much as mycobacteriosis and pseudoloma. We don't have time to talk about today. There are good drugs for treating the infections, for eradicating the eggs in the environment, etc. And so we're looking at about 10 or 12% prevalence or occurrence based on facilities. These are facilities mostly in the United States, but I'd say 10% of them are from countries from other parts of the world. Let's look at the histology. As I said, these worms invade the tissues and they cause profound inflammation, hyperplastic, dysplastic changes in the epithelium. The worms are big in relation to the intestine, so they really cause a lot of significant damage. Here's an egg here that's floating around. The eggs have a hallmark bipolar plug, so I know that's a pseudocapillaria egg. So I'm not a microbiome expert by any means. My colleague Tom Sharpton, his office is actually right next door to me, and his lab is on the same floor here in Nash Hall. This was a postdoc of his, and our team here. Here's Sean Spagnoli, myself, former lab manager of my lab, looked at longitudinal assessment of host microbiome parasite interactions resolved that occurred in the zebrafish associated with pseudoloma. And this was published in Microbiome, a top journal for the microbiome experts. And I'm not going to go through that. You've seen all these heat maps before. Big changes in the parasite burden is correlated with microbiome abundance changes. And I found this is interesting, looking at changes in the microbiome associated with worm burden. We see a statistical correlation, but it's even more pronounced when we apply it to pathology scores. So this was suggesting to us that it's not numbers of worms that are associated with alterations in the microbiome. It's actually the pathological changes to the worms. And we're going on doing a lot more work using this actually as an advantage for drug discovery of bacteria that are negatively associated with worm infections, etc., looking for novel anti-helminthic drugs from intestinal bacteria, and some other studies as well too. So we're studying the worm not only because it's a serious problem in zebrafish, but we're moving forward using it as a model to study nematode parasites in general. Okay, so the last infectious disease, if you will, that I'm going to talk about are transmissible gut tumors. So I'm calling it an infectious disease because we have shown quite unequivocally that these tumors, I'm showing a pretty aggressive one here at the junction of the esophagus and the intestine, the intestinal bulb, the anterior most aspect of the intestine, that we see these in let's say like 17, 18% of facilities. Here's a little more recent upgrade on our numbers, etc. So we see here we go, like 10% of the cases, and about 15% of the labs have been associated with this disease. So of course, tumors are going to be altering endpoints from a number of different aspects. But let's talk a little bit about these tumors. So our retrospective analysis, a student did this back 3 years ago, found that 12% of the facilities had intestinal neoplasms, 10% had nematodes. So we were looking at correlations about the worms being, that I just talked about, are these promoters for the tumors? And 5% had nematodes and neoplasm. I can show you another slide here, which is a statistically significant correlation. So basically, running simple statistics like chi-square tests, you can see that the worms, co-occurrence of the worms are beyond more than just circumstance. That actually when the worms are present, you have a higher chance of developing tumors. Along these lines, I'm going to talk about Jan Spitzbergen, who is here as well many years ago, was studying carcinogens in zebrafish and found that fish that were exposed to this direct-acting carcinogen Of the infected fish with nematodes, 50% had intestinal cancers, while 13% did not. This was statistically significant, suggesting that the worms are promoters for cancers, along these two here. We've also been looking at mycoplasma as more as a primary cause. These are associated with pre-neoplastic lesions, tumors, etc. And when you add in mycoplasm with the worm, you get more severe lesions and neoplasm. I'm going to show this here. I'm just going to get into this a little bit more. So I'm going to break this out into a few slides here. So it's just a statistical study that an undergrad did for their senior project. We published it as a note. Basically, the bottom line is, with a strong significance, is that that facilities as well as diagnostic cases were significantly correlated. The numbers of co-occurrence between the tumors and the worms was statistically significant. Keep in mind, though, we see that many times we have tumors without the worms, both in facilities as well as cases. This is telling us that the worm is probably not the primary cause. It is not the primary cause of most of these tumors, but it will exacerbate it. Again, here's Jan Spitzbergen's a little bit more data on her paper. I co-authored her with this second paper that she followed up on, showing that in this study that they didn't know the fish were infected with the nematodes. They exposed them to dimethylbenzene, anthracene, a direct-acting carcinogen, and a lot more fish that actually had worms develop these intestinal cancers. Here's another higher mag of this. She called this particular one a transmural mixed malignant neoplasm, because we're seeing all this reaction, more spindle cell reaction down here as well. Back to the mycoplasm. This is a paper that we worked with Karen Gilliman. We've shown that there is a correlation of a particular mycoplasm sequence associated with the tumors and the worm infections. And so we went back, this is without worms, and found that doing transmission studies that donor fish had the mycoplasm, recipient fish, negative control fish did not. And so these are basically group D, E, and F are recipient fish where we transmitted the tumors, but it took like 6 months, it took 9, let me go back here a little bit. To develop these tumors, it took about 9 months before we would start seeing tumors when we just exposed them to mycoplasm. And so remember, let's back up a little bit. We know that nematodes are known to be, or helminths in general, and some specific nematodes may be promoters for cancer. Like a good example would be schistosoma hematobium and bladder cancer in humans in Egypt and other North African countries. And also mycoplasm is starting to be more strongly linked with cancer. So both of these agents have been linked with cancer. So how about if we do studies, we try to disentangle this by doing what I call the field observations from zebrafish facilities and some of our transmission studies. We expose fish to mycoplasm, worms, or both. This is a paper, if you're interested in more detail, we published a couple of years ago. Here's using a in situ fish for tracking the mycoplasm. So we were able to infect them with the mycoplasm. Various examples of the mycoplasm. But these two slides are kind of basically summarizing our results. We see the inflammatory changes and hyperplasia. We would associate these as pre-neoplastic lesions in the gut. Facilities like the Stinhuber Aquatic Research Facility that does not have any evidence of these tumors do not have these underlying pre-neoplastic lesions. So we have our 4 groups. Inflammation only occurred when we had the worm present. So, but then our second endpoint, we could put this hyperplasia/dysplasia. A lot when we have mycoplasm plus PCAP, that's capillary nematode. But still a lot with just the worm, even though it was not exposed to the mycoplasm. So the worm is quite a driver of hyperplasia and dysplasia. This is where they went. The bottom line of the study was what about tumors? So we held these fish out. We couldn't get all the way out to 9 months. We got a little bit past we got out to 32 weeks, and what I'm showing here is only the groups that had the worms developed tumors. Mycoplasma plus the worms, we saw a lot of tumors. But unfortunately, just as this is the complications of the world of studying experimental disentangling the cause of cancers, a few fish, even without direct exposure to the mycoplasma, also develop the tumor. So there may be yet even another agent involved with this. Okay, so I'm pretty close to the end of what I'm going to be talking about. How am I doing on time? Can someone tell me, or is that just should I just keep talking? Okay, I know no answer, so I guess I'm okay. But I just have a few slides left. Supersaturation. Now we're switching gears. We're leaving infectious diseases, but this is a problem that we're seeing in a lot of facilities. Supersaturation is where you have total pressure of dissolved gases exceeds the atmospheric pressure, and this happens in facilities when you have water under pressure in which you have air gets into the line. Let's keep it simple like that. From a variety of causes, it can be a population level problem, but many of the fish don't develop clinical disease. Here we see air bubbles in the fish, and here we see a bubble in a vessel here, up here as well too. Bubbles form in tissues. This is due to gas emboli in the vasculature can result in occlusions and necrosis and death. The fish can be lethargic. Sometimes you don't even have the bubbles, so you wouldn't even know this. But a hallmark is the fish will go down to the bottom of the tank, even if they're not showing the bubbles. What causes it? Trapped air in airlines. I showed this picture of pumps. Leaks in pumps, heating water under pressure. But this, in zebrafish facilities, number 1 and number 3 are really the big ones. So this is from Katie Murray. She said, I used to say gas bubble disease was a rare disease that was easy to diagnose, but it's more common and more difficult to identify. So we could think of it as like the tip of the iceberg. We think that there's a lot of cases where fish are subclinically or mildly affected, where you don't have mortality, and then they go on. Here's just a couple of examples. Here's a case where the fish are dying here, etc. You can have gas bubble disease has occurred in shipping when you have them shipped in these containers where there's no air for equal liberation. And how can this happen is because when you go up in the air and the plane, the fish are shipped by air, the atmospheric pressure drops in the planes, particularly with locally operating planes where they do not adjust the air pressure in the planes. I don't have time. You can read these slides. We can go on to this more if you want. But Katie added this slide, so we have shipping. Think about underlying infections. Bianca and Luciana can tell you the headache they had with their research trying to get, because wanting to get pathogen-free fish, or if you have pathogen, doc, if you want to send your fish, many countries like Canada and South Korea, they have to be shown to be free of spring viremia. The EU wants the fish to be certified. This is viral hemorrhagic septicemia virus. They're supposed to be free of this. So these are headaches that that happen with, the challenge is if you want to receive fish from outside the country or send fish outside the country. And I just want to wrap up with this here. All these diseases we're starting to work more in may affect fish by strains differently. What I'm showing you here is a graph, some work that we're doing with Pseudocapillaria. So this is abundance mean worms per fish by group, ABs, 5Ds, TUs, and Caspers. So you look at this and go, well, particularly early on in the infection, the ABs are much more susceptible than the other lines. But early in the study, so this is 22 days out, early in the study, amongst multiple tanks, the ABs and the 5Ds suffered significant mortality due to supersaturation. So we cannot tell. So there's a possibility that these ABs as a strain or line may be more susceptible to Pseudocapillaria, or they may just be more susceptible to supersaturation, which made them more susceptible. So again, this is a problem of a confounding variable. And strain specific changes such as hepatic megalocytosis. This is a condition that we see that's probably caused by some kind of anthropogenic, some kind of toxicant. AB fish at Sorrel, at Zerk, are much more susceptible than all their other wildlife type lines. This is these enlarged hepatocytes here. It's caused by arrested cell division. What is it caused by? I'm not going to go into a lot more. This is a chronic form of it, persistent hepatic megalocytosis. We don't know the source of it. We think of this as a man-made or a natural toxicant. Could it be produced with bacteria in paramecia cultures, algal toxins? We know that microcystin, blue-green algal toxins, will cause hepatic megalocytosis in fish. So we just don't know the underlying cause of this. This is something else to be thinking about. And the list goes on. The tubigan longfin is more susceptible to background infections by bile and pancreatic duct hyperplasia. So in conclusion, zebrafish are susceptible to a range of infectious and non-infectious diseases. Some of the most common diseases have the potential to impact research, both subtle and in dramatic ways. Loss of, of course, we see losses due to morbidity and mortality, organ tissue specific pathologies. Then we have, going down the next layer, cytokine transcriptome, microbiome profile changes, behavioral changes with pseudoloma, tumor incidence is going to be altered. And then we can see another area we even needs to consider is decreased growth and secondary infections. That would be the supersaturation correlated with that. And then there are strain-specific effects. So then there's the last, if you survive all this, you got to deal with problems with shipping when you have these problems. So I'm done with my talk now. Do we have time for questions?
Thank you so much, Mike, for the excellent talk.
You're welcome.
And now let's open the question. The first one is, do you think pseudoloma could have an important impact on a study related to gene function characterization and how we can prevent pseudoloma contamination?
Well, I can't really do. I would, you could read those papers on gene function. There probably is, a transcriptome effects. I don't know. And then I think Bianca, you could tell them how to avoid contamination as well as I can, because you went through that. Well, we went through. The best way to avoid contamination is screen the fish because it's vertically transmitted. I have a whole talk on how we've developed specific pathogen-free lines here at Oregon State University. But it's basically very quickly with a PCR test, screening the broodstock and then screening a subset of the progeny. And you may have to go a couple of generations. And then Bianca wrote a whole paper on this. And then and another way to avoid it is it's much more transmitted with females than males. So if you need to rederive your line, what you would do is use wild type pathogen-free females. And I don't know if you have a population going now, Bianca, of those that you could share with colleagues so they don't have to get them from somewhere else, and then breed with the males of your line. And then you're going to have heterozygotes, and then you have to go back and cross them. That's what Bianca did, right?
Yes. We have more questions. Marcela Meireles, thank you. Regarding pseudoloma, pseudocapillaria tomentosa, sorry, what do you recommend to contain the infection? There is some medicine to control the worm in the facility?
Yes. So we have published 2 papers on this. Most of our work has been directed to using amamectin, ivermectin analog, and which they use a lot in fish. It works very well against it. And our more recent you could get my email and I can send you the papers. We found that this is incorporated in the diet, but you can also actually put it directly into the water, and it works very well for killing the worms. And then we published another paper. So you have the worms in the fish, and then you also have the eggs in the environment. And one of the things that I think is most encouraging is that if you can remove the fish, and turn the heat up in the facility to 40 degrees, that should kill the eggs. But obviously that would kill the fish. So you take the fish out and treat them, and then you could turn up the heat in the facility and kill the eggs.
Thank you, Mike. We have some more questions. Nicolas Trigueiro, he's from University of São Paulo. Are there any zebrafish genetic factors that make these worms more likely to cause tumors?
It's a very good question. I don't know that. I mean, we're just getting into more of the differences in line susceptibility, and it does seem like the ABs are quite susceptible. That's just with the worms. We have not done any research on line susceptibility or mutants, except for developing the tumors, except for Dr. Gilliman wrote a paper years ago on some knockout fish that developed tumors that were very similar to ours. So there was some evidence. And also what she did is, I can't remember the gene, but they actually inserted a gene from Helicobacter pylori into the fish, and they had a high tendency to develop the gastrointestinal cancers or gastrointestinal cancer. Are they the same tumors? I don't know. So again, if you guys send me emails separately, I can send you that paper. Neil was the first author on it. Karen Gilliman was the senior author. I should know more about it because I did the pathology on it. I was the author on it. But in that paper, they talked about the genetic alterations that they did to show fish being more susceptible to the tumors.
Have more question. Ana Lucchiari is from University of Rio Grande do Norte. Is UV light an effective sterilizing unit for zebrafish recirculant system? What else can we do to avoid contamination? And
UV light works very well against. We've done studies with a lot with pseudocapillaria and pseudoloma based on observations at the Zerk, and a few other studies we've done in my lab, that the UV used in a typical zebrafish facility would be very good. As long as the water's clean going into the UV tubes, it is very good for, very effective at killing both pseudoloma and pseudocapillaria. I don't have any information on mycobacteria. I mean, we have to go to the literature. There's a lot of the literature on UV treatment of water for mycobacteria, so I think we could just based on the information that's in literature with other mycobacteria species.
Have more questions? Yes, more one. Caterini. Thank you very much for the presentation. Regarding pseudoloma neurophilia, what do you recommend to contain the infection?
Okay, so we're doing a study. We talked about preventing it, containing the infection. This is actually a good question for that Katie Murray is dealing with right now at the Zerk, because we do have the infection going on. There's a couple of things that are going on that are in the works. One of them is that she screens populations and destroys them and does not use them for breeding because it's vertically transmitted. Corbin Schuster, who's just finishing up his postdoc there, and it was actually his PhD thesis that he did with me, developed a water test. So that's coming online. He's published his thesis and his other papers are coming out soon. It's Schuster. So once you have a water test, I think it's a PCR test where you filter water, then do PCR on it. That would be very effective. Basically, it's identifying populations and then eliminating them. The problem, the thing with pseudoloma is often it's not till the infection's gone on for 8 months or something like that do they start showing a lot of clinical disease. So you have to screen the fish or the water. You can't rely on clinical disease. And once you identify those fish, do not use them for breeding. Or at least don't use the females.
Mike, I would like to thank you for accepting the invitation to speak at your webinar.
Oh,
and
okay.
I hope to see you soon.
When are you moving to Boston?
In January.
All right. I'm flying to Boston again on Thursday.
Perfect. I will send to you my dates.
Okay, let's catch up and we'll do WhatsApp here soon, okay? I'd like to catch up with you. Are you the only one on now, or is everybody else still there?
I would like that you stay with us a little more.
Okay.
I would like to thank you very much for Tuesday's present here with us. We will be back tomorrow at 8 a.m. Brazilian time. So hope to see you all with us this holy week. And Mike, if you can wait a little bit.
Okay.
Okay.
Just a little. I need to go to another meeting here. I can like 10 more minutes, okay? Then I have to leave.
Okay. Renata would like to talk with you.
Okay.
We appreciate you taking the time today and look forward to helping you find the right tools for the right application and your endless discoveries and research. Find Science Tools has been in business for over 45 years, providing scientists, researchers, and other life science professionals with high-quality surgical instruments utilizing German steel and German craftsmanship with a secondary quality control inspection in our Heidelberg office. As we say, we only carry the best and offer a multitude of fine microsurgical instruments like spring scissors, forceps and rongeurs, but also a variety of options for wound closures and animal identification. In addition to our 3 FST offices, we have over 50 distributors worldwide. While the majority of the audience is based in the USA, please note that we can ship anywhere in the world, and any products not listed in our physical catalog or online at findscience.com can be sourced as a special order, and we will work with you to get you what you need. Before we start, for those who are unfamiliar with FST, let me go over our core values we hold ourselves to and strive to provide to our customers. Quality. Impeccable product quality is what differentiates FST from the others. Secondly, our customer support. We strive for 100% customer satisfaction. Lastly, our QC department in Germany upholds our manufacturers to the standards FST and our customers expect with every instrument.